todos y a todas, bienvenidos a este simposio del Departamento de Biología Molecular de Plantas. El día de hoy tendremos 5 extraordinarias presentaciones. Quiero agradecer a todos nuestros colegas extranjeros que hayan aceptado participar. Los temas que se tratarán el día de hoy tienen que ver, en general, revisando el programa, pues con lo que viene para esto de la biología molecular de plantas. Y esta ventana, pues hacia el futuro, es algo especialmente relevante en el marco de estos 40 años que cumple nuestro instituto, en donde estamos buscando renovarnos y estamos pues buscando expandir el impacto que tiene nuestra investigación, incorporando nuevas líneas y nuevos grupos. Aprovecharemos el día de hoy pues para conocer expandir nuestro horizonte en este campo tan importante, del cual pues creo que es el más importante de todo el instituto, porque todo el planeta depende pues de las plantas, absolutamente todo. Y básicamente pues podremos ampliar y pues seleccionar, que es también parte de lo que estamos buscando ahora, estas nuevas líneas en donde el instituto crecerá en este campo. Muchas gracias. Le paso la palabra al jefe del Departamento de Biología Molecular de Plantas, José Luis Reyes. Y por cierto, soy Laura Palomares, directora del instituto. Gracias.
Gracias, Laura. That I had in Spanish already connected, so we translate and say at the same time everything. So first of all, I'd like to welcome you, the ones that are here in the auditorium, and those that are joining us in Zoom. I think I have another window for YouTube, but I didn't check that one yet. So welcome everyone to our symposium for the Department of Plant Molecular Biology. It is a great pleasure for me to receive today all our colleagues that have gotten together from the distance. You will see the first half of the program are in Europe, the second half is in the US, and they very, very kindly accepted to attend this symposium. It is not present there, no. I think you probably saw the title of the symposium, which is our, I guess, loosely translated into English would be our roots, branches, and fruits. And when we were putting together this program, I thought of this title, and because the title tries to reflect that our guests are coming from different places, and they have been in contact with our work from many years back. So that would be our roots. They have followed us, they have worked together for a while. At the same time, there are other 2 speakers that have recently established collaborations with us, and that perhaps speaks of the growth of the department entering into different places, and that's what I consider the branches that are touching different aspects that we will hear today, some of them. Finally, there's even one of the speakers who back in time was here at the IBT. He proceeded to do his PhD outside, and then now he is a group leader. So I consider that sort of fruit of the work that has emerged from here. I think this diversity in the speakers that we will have today is a reflection of how dynamic are the activities of the members of the department, who are constantly trying to open venues for different collaborations, to symposium like this one, etc. So in closing, just not to take too much time, we hope that the talks today will allow us to continue with these contacts and that we have established over a long time. And also, I hope that they will have an impact in the development of the students who will be attending this meeting, either sitting here at the auditorium, through Zoom, or in YouTube. Okay, without further ado, I'll give the microphone to Dr. Alejandra Covarrubias, who will introduce the first speaker.
Thank you. Good morning, everyone. I'm very happy to be here and to celebrate the 40th anniversary of our institute with this symposia, with many interesting talks. And I'm also grateful to the speakers today that they accepted kindly to be with us. So to start, I want to introduce Gerardo del Toro. He is a biologist. He studied biology in the National University of Mexico, and he completed his PhD in plant molecular biology from Simposio Langevio in the laboratory of Stuart Gilmore, where he was a very good student. And after finishing his PhD, he got a long-term fellowship from the European Molecular Biology Organization, EMBO, and moved to Sweden, where he worked as postdoctoral researcher in the laboratory of Claudia Koller at the Swedish University of Agricultural Sciences in 2021, he moved together with Koller's lab to the Max Planck Institute of Molecular Plant Biophysiology in Germany. And now Gerardo has focused his research on epigenetics and plant reproduction. He has long-standing interest in understanding the mechanistic basis of imprinting and its impact on the establishment of post-zygotic reproductive barriers in plants. So I'm very grateful to Gerardo. He accepted this invitation, and I'm also happy to hear their last results and data. So thank you, Gerardo, wherever you are. I cannot see you. And so let's start.
Hey, can you hear me? I think that you can hear me, right?
Yes, we can.
Okay, perfect. Thank you very much, Alejandro, for the invitation. I am very happy here to share part of the research that I've been doing during my postdoc. And I really appreciate the invitation so I can have the opportunity to share our research. So as the title says, so I am going to present today how hybridization barriers occur in plants and how these are connected to genomic imprinting. So I think that by the end of the talk, I can convince you how important is the endosperm in establishing reproductive barriers in plants. So let's see how it goes. So to begin with, I wanted to share this scheme that is depicting how the organisms can accumulate different changes along the time, and in the end, they are isolated in reproduction. So this is basically one of the main aspects or key aspects in evolution, and this was one of the few schemes that Charles Darwin is showing in his book on the origin of the species. And if someone is interested in studying speciation, I personally think that angiosperm plants is a very good model for that, because they are very good at diversifying themselves. So we know that in specifically flowering plants, they have a very recent evolutionary story, because though they are early diverging from other group of plants, in a very short time, they were able to diversify themselves. And nowadays, we can say that around 90% of the plants that exist are angiosperms. So how is that this is happening? And so this observation was appointed by Charles Darwin back in time, saying that this was an abominable mystery. So there are different features that angiosperm plants can have that allow them to diversify to this level. And of course, they have adapted to different pollinators, and also they have developed flowers and fruits. And in connected with this, they also have they have evolved a tissue that is called the endosperm. And today I'm going to show you how the endosperm has become a battlefield for post-zygotic reproductive barriers. Of course, when we are talking about reproductive barriers in plants or in general, we can classify these in two groups. The reproductive isolation barriers that come before fertilization, that are known as pre-zygotic barriers. And these ones are established before the fertilization occurs. Basically, for instance, I can say that the plants can adapt to different pollinators. So basically, this prevents the plants to exchange genetic material. Another pre-zygotic barrier could be that some pollen are not able to germinate in other plants. So, but once that the fertilization occurs, we can talk about post-zygotic barriers. So these, in principle, the plants are able to hybridize, but the hybrid is not able to survive. And one example of that is hybrid necrosis. And another example, which is the one that I'm going to focus today, is triploid block. The triploid block was observed back in time by plant breeders because they noticed something that was very striking for them. So when you have a plant that is diploid, but you cross this diploid plant together with the identical species, but with which is tetraploid, they observed that the progeny was aborting. And this was particularly striking because in principle, they are the same species. They only differ in chromosome number. And they observed that the hybrid was not able to form. The seeds were aborting. And these are known as interploidy crosses. Something that is interesting as well is that breeders also noticed that this phenotype was reminiscent to the one that they were observing when they were crossing different species. So interspecific crosses were showing the same phenotype. They were able to fertilize, they were able to mate, but the progeny was not able to develop. So triploid block is a very important speciation mechanism because it's a very early acting barrier. And this is because in nature, we can observe that polyploidy is very commonly observed. And this is due to the unreduced gamete formation or polypersemy. So when we have a population of diploid plants, the tetraploids can spontaneously appear in the population, and they immediately have an isolation barrier with the diploid cousins. And this can potentially lead to a new species. So that's why triploid block is a very important study because it can act as a very early acting reproductive barrier. So how is that this happens? I mean, what is really going on inside the seed when you have an interploidy cross? So normally, if we look inside the seeds, we notice that, I'm going to show you later in more detail, but this is an embryo in Arabidopsis that it's forming the cotyledons here, and then it's forming an endosperm around. And these endosperm, which are these cells that are cellularizing around the embryo, look healthy because endosperm cellularization is a very important developmental stage for the seed for reasons that are not fully understood. But in interploidy crosses, what we notice is that the endosperm fails to develop and specifically fails to cellularize. So this was one of the first observations that tell us that the endosperm is the one that is potentially being affected in these interploidy crosses. And this is also supported by the fact that if you isolate the embryo, you are able to grow the triploids plant, meaning that the endosperm is basically causing the embryo rest, and then obviously causing the seed abortion. Interestingly, when we look at interspecific crosses, we notice a very similar phenotype. As well, the embryo is forming, but at some point it stops developing, and the endosperm also is not cellularized, meaning that interploidy and interspecific crosses share a lot of things in common, meaning that they may have a very similar mechanism behind. So just to bring everyone to the same page, maybe for this audience, it's not that necessary to explain in further detail, but just to show you very quickly how the endosperm develops. So in principle, the pollen grain germinates and is releasing two sperm cells. One of the sperm cells is going to fertilize the egg cell, and the other one is going to fertilize the central cell. giving rise to the embryo and to the endosperm. Particularity is that once that the endosperm is established and is developing, it has a nuclear phase. In the case of Arabidopsis, most of the flowering times have this kind of developing endosperm. And after a set of nuclei divisions, the cellularization starts. And this is a step that I was telling you that fails to be established in triploid seeds. Something that is interesting about the endosperm that I don't know you have noticed is that the endosperm is the product of a second fertilization from the central cell. And the central cell is homodiploid, meaning that it contains 2 copies from the maternal genome. So after fertilization, the endosperm has 2 copies from the maternal genome and 1 copy from the paternal genome. So, and this is important because when you mess with this balance, then you affect seed development and in specifically the endosperm development. But how is that this is happening? And something also very striking about this is that whenever you have a cross between a tetraploid and a diploid, the phenotype that you observe depends on in which direction you are using the tetraploid parent. When you have a normal plant, as I was explaining before, the endosperm develops nicely, cellularized, and the seed development can continue. But if you are using a tetraploid plant as a maternal donor, what you have is a maternal excess parent, a maternal excess cross. And this affects the endosperm in a different way. In this case, the endosperm cellularization starts earlier. And in comparison, when you have a tetraploid paternal plant pollinating a diploid plant, you have a paternal excess in the endosperm, and in this case, the endosperm is delayed in cellularization. So these non-reciprocal phenotypes that are being observed whenever you have an interploid cross are connected somehow to genomic imprinting. And breeders before they have described this phenomenon, and they have established a concept that is known as the endosperm balance number, meaning that the 2-to-1 ratio of maternal and paternal genomes in the endosperm is key to have a proper endosperm development in the seed. So, but how can we study this? So we are interested to know which are the molecular mechanisms behind the triploid block. And working with tetraploids, as you can assume, is not that easy, especially if you want to do genetics. But we are using Arabidopsis as a model to study this, and we have a lot of tools. And one of those tools is to use mutants that are affected in meiosis, meaning that in some mutants like OSD1, which stands for omission of second division, and JSON, which is another mutant, they are affected during meiosis in a way that the phenotype of these mutants is that they form unreduced gametes, meaning that you can mimic the phenotype, or you can mimic the tetraploid if you are using these mutants. So in this case, I am showing you how if you are using a mutant that is for OSD1, you can, as a paternal donor, you can mimic the paternal excess cross, meaning that we can make use of these mutants in order to study the genetics behind the triploid block, which is what we are interested in. And one of the early works in this kind of studies trying to elucidate the genetic basis of the triploid block used JSON, which is one of those mutants that I'm telling you that is affected in meiosis and produces unreduced gametes. And in a mutant screening, they found a set of mutants that are able to suppress the triploid block. One of those mutants was admitos. They call it admitos. So in the double mutant background of JSON and admitos, you have a suppression of the triploid block phenotype. So if you compare to the JSON single mutant, you have abortion, which is reminiscent to the triploid block, causing the endosperm to not cellularize, and also to causing the embryo to arrest. But in the double mutant with admetos, you have that the endosperm cellularization is reestablished, and you can form a proper triploid seed. So this was the first effort in order to develop or to understand the genetic basis of the triploid block. And along the years, Claudia's group have described a number of mutants, a number of genes that are involved in the establishment of the triploid block. And there is a particularity about them. They are meaning that all the suppressors that we know so far. So in this graph, I'm just showing how in mutant backgrounds of different of these genes, so you are able to establish triploid seeds. They suppress the triploid block, and all of them are paternally expressed genes, meaning that genomic imprinting is directly involved in the establishment of the triploid block. So one of the key questions that we are interested is to understand how genomic imprinting is established in the first place, and also how can we discover more imprinted genes so we can understand better what is going on with the triploid block. So just to explain better how imprinting works, because I'm not sure if everyone is familiar with this. So the genomic imprinting is established in the endosperm, and this is an epigenetic phenomenon that causes that, depending on the parent of origin of certain alleles, the maternal alleles are repressed, or the paternal alleles are repressed. So normally in most of the genes in endosperm are biallelic, meaning that they are expressed both from the maternal and from the paternal copies. But what it happens with the genomic imprinting is that you have two different kinds of expression. You have maternally expressed genes, and you have paternally expressed genes. And what happened here is that in maternally expressed genes, the paternal allele is completely repressed, meaning that only the maternal alleles are being expressed. And it is the opposite way when we are talking about paternally expressed genes. But how is it this phenomenon is established during reproduction? Well, we have a bit of an idea. What happens, at least when it comes to maternally expressed genes, is that there is an enzyme which is a DNA glycosylase that is specifically expressed in the central cell. This is before fertilization, and then this demethylates the maternal copies of the genome, meaning that after fertilization you will have a demethylation of the maternal alleles, so they can express. And this is not happening in the paternal genome, meaning that the paternal alleles are being repressed. And when it comes to paternally expressed genes, what we know is that H3K27 demethylation, which is a repressive mark, is connected to the repression of the maternal alleles. And then this allows the expression of the paternal alleles. So during this talk, we are particularly interested in paternally expressed genes because most of the suppressors of the triploid block, as I was mentioning before, are paternally expressed genes. So just to show you a model that we have right now, along the talk I'm going to try to complete. We know that the polycomb complex, which is responsible for establishing repressive marks for H3K27 demethylation, is expressed before fertilization, and these repress the maternal alleles. And this phenomenon is not occurring in the pollen, allowing those genes to be expressed from the paternal side. So one of the main reasons we are interested in the epigenetic regulation of these genes is because we would like to know how many imprinted genes we have in Arabidopsis and how they are connected to the triploid block. And that was one of the first things that I was doing during my postdoc. And of course, studying endosperm is not that easy because this is we are talking about tissue that is inside the seeds, so it's very hard to reach it. And what previously former postdoc in the lab, what he did was to establish a transgenic line that allows isolation of endosperm nuclei. And this is because this transgenic line has a nuclear protein that is has a domain that can be biotinylated, and at the same time it has GFP, and at the same time we have transgenic expression of a biotin ligase. And when you have these two components in the same tissue, what you have is that the nuclei are biotinylated, and you can isolate the nuclei using streptavidin in order to isolate the nuclei from the endosperm. So he established this transgenic line, and I was using it to isolate endosperm nuclei and to perform, to begin with, a transcriptome in order to see how many imprinted genes we have in Arabidopsis. So, as I was mentioning before, this is particularly challenging because it's a tissue that is inside the seed. And of course, we don't want to have contamination from the maternal tissues around, because then you won't be able to ask which genes are maternally expressed and paternally expressed without having this contamination. It's actually very difficult. Previously, a lot of efforts have been made in order to establish parent of origin expression in seeds. And in this figure, I am showing you that an enrichment of different tissues, embryo, suspensor, endosperm, and seed coat in those transcriptomes. And indeed, those previous transcriptomes have enrichment of endosperm tissues, as you can see here, in yellow and orange color, but they also have enrichment of seed coat. So this prevents you to have a better overview about the imprinting expressed genes. So by using intact, so I established this transcriptome, and indeed I do have enrichment of endosperm tissues and very less contamination from maternal tissue. So this technique allows us to have a better resolution when it comes to imprinted genes. So what I observed in this transcriptome, that I did, was that indeed most of the genes are biallelic, but there is a group of genes that are either maternally expressed in orange or paternally expressed in blue. And something that was striking, because this is the first transcriptome that is published at 4 days after pollination, because also imprinting may have variation depending on which stage you are looking at. In this case, we are observing 4 days, which is the earliest point that has been observed so far. So we observe a higher number of paternally expressed genes. So because previous transcriptomes have reported less, which might be due to the fact that maybe in earlier stages of the endosperm, you have more imprinted genes, or just because the quality of the data is better. But so we have now a set of new paternally expressed genes, and we can have more confidence in to call these imprinted genes indeed imprinted. So of course, we also have a percentage of maternally expressed genes, but as I was saying before, most of the genes are biallelic. So, but what we can do with this data? So first of all, we notice that indeed many of the suppressors of the triploid block were present in this group, but also we are interested how these genes are regulated at the epigenetic level. And using the same intact or the same technique, a former postdoc in the lab also noticed something very particular about these paternally expressed genes. So as I was saying, the repressive mark of K27 trimethylation was previously known to be associated with the repression of the maternal alleles, as you can see here. So this is a group of PECs, and they have definitely an enrichment of K27 trimethylation. And this is in pink, you can see the maternal allele in comparison to the paternal allele. So this repression is specifically present in the maternal allele. But also another observation that was striking was that these genes also show a very high enrichment of K9 demethylation, meaning that these two repressive marks are simultaneously repressing the maternal allele of PECs. And this was striking because these repressive marks are normally not connected to each other. They they are exclusive repressive marks. So it was particularly interesting to see that the repression of the maternal alleles in PECs is accomplished by these two very uncommonly seen epigenetic marks at the same loci. And H3K9 dimethylation is also associated with a CHG DNA methylation. And indeed, also the PECs have a very high accumulation on the maternal alleles of this repressive mark. So we have a scenario where we have a number of PECs that are being repressed simultaneously by these epigenetic marks. So we were wondering if by using these three epigenetic marks, we can predict which genes are going to be imprinted. So using the transcriptome that I was describing before, we organized the genes in different levels from 0 to 12, being 12 the group of genes with the highest scores for co-localization of K27, K9, and CHG methylation. And indeed, these genes that are in this group are more paternally expressed than the rest of the groups, meaning that when we take a look to the expression of these genes that co-localize all these repressive marks, most of them are indeed paternally expressed. So this way we can add to this model that not only K27 trimethylation is repressing the maternal allele of PECs, but also at the same time we have K9 demethylation and CHG methylation, which adds to our knowledge about how imprinting is being regulated. And interestingly, we have more PECs that have been described before, meaning that these imprinted genes might be potentially fast evolving. And if you want to know a bit more about how this particularity of epigenetic marks co-occur in these genes, I can refer you to this new finding that has been published very recently in the lab, where a former postdoc found that indeed these three repressive marks are important to co-occur because these repressive marks are present in genes that are involved in seed dormancy. So we know that REF6 is a demethylase that removes K27 from the system, allowing expression. But if you have K9 demethylation, you have that these loci are protected, and this is important because many genes that are repressing seed germination are marked with these three epigenetic marks, meaning that also the seed germination is regulated by imprinted genes. So as a summary for this first part, I'm just mentioning again that indeed imprinting is connected to hybridization barriers, how we have been observed. And we have now a set of maternally expressed genes, paternally expressed genes that we can study more in detail how these are regulated. And we know that at least these three repressive marks co-localize for many of them. But now I would like to give you an example about how is a triplet block being established by one of those paternally expressed genes. In this case, I'm going to focus in PEC2. So PEC2 was one of those genes that was found that when it is mutated, it represses the triploid block, showing that a very, very high proportion of seed germination in triploid seeds, meaning that it suppresses the triploid block. So, but previously also we show that PEC2 is highly expressed in triploid seeds. So here, this is just qPCR analysis showing how PEC2 is overexpressed in triploid seeds in comparison to diploid seeds. And something very interesting about this gene is that even though it is upregulated in triploid seeds, as you can see here in this qPCR, a translational reporter line doesn't show any translation, meaning that it might be the mRNA and not the protein, the one involved in the function of PEC2 in establishing the triploid block. So we show in this paper that, just long story short, that PEC2 actually has a target sequence for a small RNA that we know as small RNA A54. And what we described before is that PEC2 is overexpressed in triploid seeds, and it works as an sponge to sequester this small RNA. And basically, this depletes this small RNA from the system and prevents this small RNA to bind natural targets like UBP1B. So I'm not going to go more in detail about this aspect, but I'm going to show you more of how this PEC2 is potentially being conserved in different accessions in Arabidopsis, because we were interested how this sequence that is targeted by this small RNA in PEC2 is conserved in different accessions. And I started this project with my former master student, Varsha Vasudevan, which we worked together in order to see how this small RNA target sequence in PEC2 is conserved in different accessions. I'm not showing the data, full data here, but most of the accessions contain the target sequence for this small RNA almost intact, but there is a group of accessions that have all sorts of indels, either deletions or insertions specifically in the area where this small RNA is binding. So we found this is a striking variation, and we were wondering how this could be connected to the triploid block. In silico predictions show that even though all these accessions might have these insertions or deletions, they might potentially bind to the small RNA, even though they have this variation. So, but then we wanted to see if this was the case, and specifically to begin with, how these mutations in the PEC2 gene affect the triploid block in all these accessions. So what we did was to first establish tetraploid lines for all those accessions. So that's something that we can easily do in the laboratory using colchicine. And after we tested the triploid block. So here in this picture, I'm showing you just an overview how the triploid block looks in different accessions. And if we put this in numbers, indeed, something that was very surprising for us was that the seed abortion, or let's say the triploid block phenotype, varies in all the accessions, regardless of how conserved the sequence for PEC2 is or not. Like, for instance, here in Colombia, an RLD have identical PEC2 sequence and identical target sequence for this small RNA, but yet they are showing a very different triploid block phenotype. Here, for instance, if you focus in fully collapsed seeds, RLD has around less than 20%, and Colombia has more than 60%, meaning that though they have the same sequence for PEC2, they show very different phenotype for the triploid block. So then we were wondering how is that this can be explained if it is not because of the variation of the PEC2 sequence. So if we consider that the expression of PEC2 might be relevant for this, because if you remember, PEC2 is working as a sponge for this small RNA. So the highest expression you have, potentially the higher the sponge effect is going to be. So then we were looking at the expression of PEC2 in all those accessions, in those triploid seeds. And what we noticed was that those accessions with the highest triploid block phenotype, like Colombia, Crow, and Ban, have the highest expression of PEC2. And this connects with the fact that indeed, if you have a higher expression for this sponge, for this small RNA, you can deplete more efficiently these small RNAs from the system. And in theory, this might be connected to the triploid block phenotype that we are observing. So if we do a correlation about seed abortion and expression, so we can indeed notice that the correlation between the expression of PEC2 and the abortion is highly positive. So when you have a higher seed abortion, you have a higher expression of PEC2. So, but then we would like to see if though these different accessions have mutations in the PEC2 gene, we are interested to see how that is connected to the triploid block. So we don't know, even though we have in silico predictions that PEC2 is still being targeted by this small RNA regardless of the mutations. Of course, we need to test this in a more functional way. And to do this in the best way possible, what Barsh established was these CRISPR mutants. So what we did was to design an experiment in order to have two different mutants in PEC2. One mutant that removes the part of the gene that contains the target sequence for this small RNA, and another mutant that removes part of the gene, but after the target sequence for this gene. So in this case, we thought that by having this set up for the experiments, we can use PEC2 from all these accessions in order to complement the mutation of PEC2. So, of course, maybe you are wondering how these mutants behave when it's connected to the triploid block. We know that PEC2 suppresses the triploid block, and that this function is connected to the binding or the sponge effect that it has against this small RNA A54. And indeed, when we tested the triploid block in these mutants, you can focus in this mutant, for instance, PEC2-2, which is the one that removes the target sequence for this small RNA. Indeed, this one shows a lower seed abortion phenotype, meaning that indeed this mutant is able to suppress the triploid block. And it's not the same for this other mutant that has the deletion after the target sequence for this small RNA. This mutant is not able to suppress the triploid block, showing seed abortion to a similar level as a wild-type scenario, confirming again that indeed this target sequence is relevant for the function of PEC2 in establishing the triploid block. And now the question is how the variants from all those accessions that we have now is connected to the triploid block, and this is something that is in work in progress. As a summary for this second part, just to mention that indeed PEC2 is a gene that is highly conserved well, I didn't show the data in different species, but also especially in different accessions from Arabidopsis. The target sequence for this small RNA, it's likely conserved. We have to test yet in silico if those copies are functional, but we have a correlation that the transcript level of PEC2 is correlated with the triploid block phenotype. So I hope that along the talk, I show you that now we have a better understanding how imprinting occurs, how paternally expressed genes are being regulated by the colocalization of these three repressive marks, by getting knowledge about how many PECs we have in Arabidopsis and how they are connected to the triploid block, and also specifically in the paternally excess cross. One of the mechanisms that we know that is connected to this phenotype is PEC2, which works as a sponge, and we are now trying to understand how is evolutionary conserved or not in different accessions in Arabidopsis. Finally, I would like to thank you for the invitation again, and also for the attention. Definitely, it has been challenging to do all this work with pandemic and moving, but before we were in Sweden, as Alejandro was saying, now we are in the Max Planck Institute in Germany. And I would like to thank also my main mentor, which is Claudia Koller, and also my former master student, Barsha, which we were working together in the PEC2 project. So if you have questions, please. Thank you for your attention.
Thank you, Gerardo. I am not a plant scientist, and the presentation was very clear. Thank you very much. My question is regarding the viable seeds that you have. I mean, you show us that even in the worst case, there's 80% of seeds that are not viable, but how the seeds that are viable in those conditions remain viable? What's their characteristic? Thank you.
So yeah, the phenotype definitely is not fully penetrant, let's say. And this varies a lot. For instance, in one of the examples that I was showing you was this crow accession from Arabidopsis. And in those, we find like the phenotype is more than 90%, more than 90% of the seeds abort. And of course, you always find variation. Some of the seeds manage to sort out the triploid block, though they might not germinate. Sometimes that's why we also test seed abortion and also germination, because though they might look fine, when we test germination, they don't germinate fully. And of course, you have a very few percentage of those that manage to germinate, which is the less of them, and they will form a triploid plant. But they are mostly sterile.
Hi, Gerardo. Thank you very much for your talk. It was very pretty. And I have two questions. One is there any function of the microRNA 854 outside of the endosperm?
You know that this small RNA binds to UBP1B, which is a component of stress granules. And this has been shown to be the case in vegetative cases. But also this small RNA is very particular. It doesn't express in a very high level. And the expression of this small RNA in vegetative tissues is ectopic, meaning that only when transposable elements, specifically Attila-6 transposable element, express or gets activated in mutants like DDM1 or MET1 that are mutants that are lacking CG methylation, these transposons reactivate, and then this small RNA is synthesized after this reactivation of transposons. So, so the answer is yes, it has a role, but whenever you have reactivation of transposable elements, and this small RNA regulates components specifically for the stress granules, so like UBP1B. So it might work in that scenario outside the endosperm.
So is the mutant viable of the mutation of the small RNA? Do you know if there is?
So it's we don't have mutants for this small RNA because this small RNA is derived from transposable elements, and at least for this specific small RNA, there are at least 12 different loci that can generate this small RNA. So as far as I know, there is actually no mutant on this small RNA, and it might be very challenging to do that.
Okay, yeah. Thank you. And my second question is the PEG2 function, because you are clearly showing that is the RNA that works as a sponge, but is the protein have any function, or it is expressed somewhere in the embryo, or it's only expressed in the endosperm?
So this gene is only expressed in endosperm, but also we have seen expression in the sperm cells. We don't know why it's expressed in the sperm cells in the pollen. So the protein has no function that is known, because there is no domain that we can see in the protein that can give us a little bit of idea what the protein can do. So we don't have an answer for that. But what, at least when it comes to the triploid block, the mRNA is the one that has a function.
Hi, Gerardo, very nice talk. I have a question. With your intact RNA-seq, do you see difference in the different domain in terms of imprinting, in terms of the different domain of the endosperm?
Yeah. So yeah, we haven't checked that in detail, but our transgenic line has a slightly stronger expression in the chalaza endosperm. So meaning that we might have also more imprinting genes because we are specifically or mostly sorting or selecting nuclei from the chalaza endosperm. So it might be. So there is a paper recently published by Gerin's group where they are looking at imprinting at different domains in the endosperm, and there is variation indeed. But we haven't looked at that because our line expresses all over the endosperm. So it's like using the intact approach. It's a bit difficult to distinguish different endosperm domains. But yeah, we might have an enrichment of a chalaza endosperm.
Thank you.
Gerardo, I have the last question, I think. You said that the over-methylation in the embryo is a restriction for germination, right?
So what's happening is that some genes like AB3 has these 3 epigenetic marks. So this AB3 has been connected to represent germination. So you want to well, at least what we know is that if you keep these 3 epigenetic marks representing AB3, for instance, you can allow germination to happen. So mutants that are that are defective in K9 demethylation show dormancy. So connecting methylation with demethylation with dormancy. Indeed. Yeah.
Yeah. So it's modulated by abscisic acid then.
Exactly.
Okay. So thank you so much, Gerardo, for this very interesting talk, and see you soon, I hope. Good luck with your work. Thank you.
Thank you very much for the invitation. See you.
So our next speaker will be Dr. Julie Gray. She's already connected. Oh, okay, Julie. I cannot see you, but I don't know if I have to stand here. So I'm also very, very happy that Julie Gray accepted this invitation. I know her well, and she's a great person and a great scientist. So the introduction for you is that Julie Gray studied biochemistry at Liverpool University in the UK and was a PhD student at the Nottingham University, where she researched genes that control fruit ripening in the team who created slow ripening GM tomato products. She won Royal Society funding to carry out postdoctoral work in Australia at the University of Melbourne, where she investigated how flowers prevent self-pollination. Now, Julie Gray is professor of plant signaling, of cell plant signaling at the University of Sheffield, and her group study stomatal development and function. Actually, most of the very good contributions to plant science have been on this topic particularly. Recently, they have created crop with, I think with Julie's group, have created crops with fewer stomata that have improved drought tolerance and reduced water requirement. Her group works on Mexican beans with UNAM and Caspar Chatter. Well, you know that we won the Newton Prize in 2018. And she also studies the signaling pathways that allow stomata to open and close, and has shown that these pathways evolved soon after the appearance of stomata in the fossil record around 400 million years ago. Julie now is member of the European Molecular Biology Organization and is currently enjoying study funded by a Royal Society Liverpool Senior Fellowship. I don't know if I pronounced it well, but with that, I leave you with Julie. Thank you very much, Julie, and hello. I am around here. You see me.
Hello. I'll try and share my screen. Thank you very much for that very kind introduction, Alejandra, and hello from Sheffield. Good afternoon from Sheffield in the UK. So it's a real pleasure to be invited today. thank you so much for letting me be part of your celebration, and congratulations on your 40 years. I'd like to start by thanking the people whose work I'm going to talk about today. In my lab, mostly I'm going to talk about gene discovery work by Lee Hunt, and the rice work is mostly done by Bobby Kane, with collaborators in the Philippines, Paul Quick and his group at the International Rice Research Center, and Apachar Vanavicit and his group at Kazakhstan University in Thailand. And I'd also like to say a really big thank you to all our collaborators in Cuernavaca, where Casper Chater has been working and collaborating for about the last 7 years on various topics to do with the stomata in beans. So Miles and Jessica have been working on beans in my lab, but we have lots of wonderful collaborators in Mexico. So I'd like to say a big thank you to all of them, and particularly to Alejandra, who has been so unstinting in her support of our projects and of Casper for so many years. So I'm not going to talk much about soybean today because it's mostly led by Casper. I'll just show you one slide at the end of what we're doing on that. So I'm a stomatal biologist. I'm interested in how stomata open and close and how stomata develop. So stomata are pores on the leaf surface, and you can see in this leaf cross-section that they're important for allowing carbon dioxide into the leaf through the waterproof cuticle so that it can access the mesophyll photosynthetic apparatus where the carbon is fixed. And while the stomata are open, they also allow water to escape from the plant in the transpiration stream. So stomata have both advantages and disadvantages. Their major advantage is that they allow carbon into the plant for photosynthesis, and they also allow cooling of the leaves through water loss and pull up nutrients in the transpiration stream. But the downside is that they lose a lot of water, that for every carbon dioxide molecule that's fixed, we estimate that maybe 100 or 200 molecules of water are lost. So what I want to think about today is whether we could improve that ratio. We're living in times of rising carbon dioxide levels, that CO2 levels have gone up by over 40% since the Industrial Revolution. So plants are not starved for carbon. Could they get by with less stomatal capacity and then save a bit of water? Because under climate change, as our temperatures are rising, more water has been held in the atmosphere, which means there's less water in the soil available for agriculture. And we cannot keep irrigating our crops. We don't have enough water to do this. So we have to make crops that are more water use efficient. That is, that they fix more carbon per water molecule lost. Another downside of having stomata is that they're nice convenient entry holes for many pathogens that get into the plant, some of them just through these stomatal portals. And so because of these downsides, plants have evolved the capacity to open and shut their stomata so that they're only open when they need them. So this video shows wheat stomata shutting. It's speeded up. It takes about half an hour for them to shut. And stomata close in the dark to conserve water when they're not photosynthesizing. They also close when they receive drought signals such as abscisic acid so that they can conserve water when there are limited resources. and they can also close in response to some pathogen signals. So what I want to look at is the idea of whether you could make plants with fewer stomata that would be better suited to our current high carbon dioxide environment and low water availability environments. So my hypothesis that I want to look at with you today is that plants will need fewer stomata to maintain their photosynthesis at current and future CO2 levels, which are so high. And this would mean that they could use less of our precious water resources and perhaps have improved drought tolerance. So if we could make plants like the picture on the right, perhaps they would need less water. And less water is important because of our drying soils. Even in the Philippines, where I collaborate with people and we think about agricultural system being tropical, they are having major droughts that are affecting rice yields. It's estimated that 34% of rice is currently lost to drought, perhaps 40% in India. And I know that Mexico is also experiencing drought and it's affecting crops more and more. So if we could do this, it could be advantageous. And that's what I want to explore in my talk today. So my plan is to talk about how stomatal development is regulated, what we know from Arabidopsis, and whether we can use that knowledge to manipulate stomata to produce crops that perhaps would use less water. And if we did that, what else might we affect? Would we affect pathogen tolerance or nutrient uptake or all sorts of things? So we know from many studies that stomata develop when the leaves are very tiny. In Arabidopsis, they're developing in the center of the rosette. In leaves that are just perhaps a couple of centimeters long, they're patterning their epidermal cells. And there are dispersed cells throughout the epidermis that are capable to undergo an asymmetric division to produce a smaller daughter cell that we call a meristomoid and a larger daughter cell that usually goes on to become a pavement cell. This meristomoid will continue to divide asymmetrically several times until it's surrounded by its own daughter cells, and then it will differentiate to form a guard mother cell that will undergo a symmetric division to produce the two guard cells that mature to form the stomata and the pore. Now, some of these pavement cells might also maintain meristomatic fate and produce secondary guard cells. So this cell division pattern helps to space out stomata on the leaf surface. We were interested in looking at genes that are expressed in stomata and during stomatal development, and we came across this epidermal patterning factor that is expressed just in these meristomoids very early in epidermal development. By the time the two guard cells have formed, it has disappeared. So it's only expressed in these very early stomatal precursors. It's a secreted signaling peptide that prevents the surrounding cells from entering the stomatal lineage. So it helps to space out the stomata on the leaf surface during development. It's sending out a signal to say, I'm going to develop into a guard mother cell, so you surrounding cells can't. So it's an inhibitor of stomatal development. And we know that because when it's knocked out in these two tDNA insertion lines that don't express the epidermal patterning factor, they produce more stomata, an increased number of stomata on the leaf surface. And when we overexpress this epidermal patterning factor by introducing another copy of the epidermal patterning factor gene on a strong promoter, in this case, the cauliflower mosaic virus promoter, we make plants that have fewer stomata. This Arabidopsis line only has 20% of the normal number of stomata. And this is what I'm going to be talking about. These plants that are overexpressing an inhibitor of stomatal development, and they produce fewer stomata. So in the background, you can see I've marked with red dots the stomatal pores, and in the plants that are overexpressing this epidermal patterning factor, there are far few stomata being formed. So this epidermal patterning factor that I've put here, and it's relative in pink and orange, bind to the erector receptor in the neighboring cells in the plasma membrane, and they activate a phosphorylation signaling cascade that eventually phosphorylates a transcription factor known as speechless. Normally, speechless promotes cells entering the stomatal development pathway and progressing through the stomatal development pathway. So normally speechless is directing stomatal development, but when it's phosphorylated, it's destabilized and inhibited. So when this epidermal patterning factor pathway is triggered, it inhibits the production of stomata. Now, it's a little bit more complicated in that there is a second epidermal patterning factor like signaling peptide that can also bind to the same receptor, but it doesn't activate it, it blocks it. So whereas the original epidermal patterning factor that I told you about inhibits stomatal development, this epidermal patterning factor like peptide promotes stomatal development. And so it is the balance of these positive and negative antagonizing signals that determine how many stomata will develop on the leaf surface. So through manipulating these genes in Arabidopsis, we produced a range of Arabidopsis plants with from just 20% of the normal number of stomata up to over 3 times the normal number of stomata. And we can tell that these stomata were functional because when we look at them with an infrared camera, a thermal imaging camera, to look at their temperature, we can see that the plants with fewer stomata are a couple of degrees warmer than the plants that have more stomata. And what we're looking at here is the evaporative cooling produced by the water loss through the stomata. So these plants with more stomata are losing more water, and that is cooling their leaves through evaporation. And in this false color image, you can see the cooler colors are these dark blue colors, and the plants with fewer stomata are losing less water. So just looking at from now on, just looking at the epidermal patterning factor overexpressor and knockout here, and comparing them using infrared gas analysis to quantify the gases going into and out of a leaf cuvette, we can measure firstly on this graph, the water loss from the leaves, and see that the plants that had only 20% of the normal number of stomata have about 45% of the normal amount of water loss. So they have a substantial reduction in water loss, whereas having twice as many stomata gives you enhanced water loss. So quite big changes in water loss from the leaves of these plants. Looking at carbon dioxide assimilation, we have a similar trend, but it's nowhere near as big a reduction. So although these plants only have 20% of the normal number of stomata, they are assimilating about 80% of the normal amount of carbon. So they have a small drop in photosynthesis, whereas having more stomata doesn't give you much advantage. And I think this is because the stomata can still open and close. So they're only taking up as much carbon as they need. So a big drop in these low stomatal density plants in water loss and a smaller drop in photosynthesis, which means that these plants are more water use efficient, that they can assimilate more molecules of carbon for molecules of water lost. So they have increased water use efficiency when they have fewer stomata. So what does that mean? Does it mean that they're drought tolerant? Well, the plants grow entirely normally and go through their life cycle, and they are drought tolerant. So in this experiment where we've just not watered the Arabidopsis plants for 10 days and then rewatered them, only the plants with fewer stomata that were overexpressing this epidermal patterning factor survived. So they have both enhanced water use efficiency and enhanced drought tolerance. What this meant is that if we weigh the pots during the drought experiment, we can look at how much water is left in the plant and the soil. And the plants with low stomatal density maintain a higher weight. That is, they have more water in their soil and in the plants. And by 6 days without watering, you can see that the amount of water left in the soil is about 40% greater than the wild type. So they're better at preserving soil moisture. We can also look at when the stomata close, looking at stomatal conductance. We can see that the plants with a high number of stomata, with a high stomatal density, start off with a higher stomatal conductance, and after 3 days they have to shut their stomata because they've run out of water. They're getting a drought signal and they're shutting. The wild type plants run out of water after 4 days and shut their stomata, but the low stomatal density plants that have more modest stomatal conductance levels don't run out of water and have to shut their stomata until after day 8. So they're better at conserving water, and they also have this window of opportunity here for several days when their stomata are still open and they're still able to photosynthesize, they're still able to cool, they're still able to pull up nutrients because they haven't shut their stomata. So potentially the low stomatal density plants, albeit having a slightly lower photosynthetic level, could have advantages under drought conditions. So what about disease resistance? We looked at Pseudomonas syringae, bacterium that gets in exclusively through the stomatal pores in Arabidopsis and causes bacterial speck disease. We can see that if we sprayed the plants with Pseudomonas syringae and then ground up the leaves and looked at how many colony-forming units got inside the leaves, after 4 hours, there were far fewer bacteria in accessing the interior of the leaf in the genotypes with a low number of stomata than the wild type and the ones with higher numbers of stomata. And we could look at damage using fluorescence imaging to look at the activity of photosystem 2. So this graph is looking at the area of the rosette that is damaged by the pathogen. These two lines at the bottom are a mock inoculation, so they're showing no damage at all. The top line in black are the wild type plants showing increasing levels of damage to photosystem 2. So this is biochemical damage caused by the drought up to 2 days. whereas the plants with fewer stomata are showing a much smaller area of damaged leaf tissue. So for several days, they're showing tolerance to this infection. So they have an increased level of pre-invasive and post-invasive immunity. So hopefully I've shown you that stomatal density, changing stomatal density, can influence disease resistance, drought tolerance, and soil water conservation. And plants with fewer stomata enhance all of these that are important crop traits. So I was really interested in seeing if we could carry out similar stomatal manipulations in crop plants. And I particularly wanted to look in cereal plants because wheat and rice use a lot of water in their agriculture. It takes perhaps several thousand liters of water to produce a kilogram of wheat or rice grain. So they're not at all water use efficient. However, the grasses have quite different stomatal morphology. They don't have these kidney-shaped guard cells. They have dumbbell-shaped guard cells and subsidiary cells, and the stomata form in these parallel leaf files. However, with the help of collaborators, we managed to find epidermal patterning factors in the barley, wheat, and rice genomes. With the James Hutton Institute, we overexpressed the barley epidermal patterning factor, and with the NIAB in Cambridge, we overexpressed the wheat epidermal patterning factor. And again, we did both of these on strong promoters, on actin and ubiquitin promoters, and this produced a reduction in the number of stomata that formed. As you can see, the plants had a degree of drought tolerance. These are plants that have about half as many stomata in a drought experiment in both the barley and the wheat. So they are less wilty under drought conditions. Again, looking at damage to the photosynthetic apparatus caused by the drought. This is just looking at spot measurements. You can see that the photosystem 2 efficiency falls off much more quickly in the control plants. After 3 days of drought, they're starting to suffer, whereas these 3 wheat transgenic lines are holding up much better through the drought. So some degree of drought tolerance again. I've done most of our work in rice, so I want to look a little bit more detail at our rice experiments with you. So these are our rice plants overexpressing the epidermal patterning factor, growing at the International Rice Research Institute in a screen house that just has nets between the outside environment. And these people down the edge are all measuring stomatal conductance with infrared gas analysis. We looked in detail at 2 lines, one that had just less than half the normal number of stomata on its leaf surface, on leaf 5, and I'm calling this low stomatal density. It's sort of similar to the barley and wheat that I showed you, but less severe than the Arabidopsis line that I showed you. And we also looked in detail at a very low stomatal density line that has just 12% of the normal number of stomata. So this is more severe than the Arabidopsis line. And remarkably, it goes through its life cycle normally, looks apparently normal. The infrared gas analysis shows that both of these have substantial reductions in their water loss. This one, with about half the number of stomata, had no significant drop in carbon assimilation or photosynthesis. This one, with only 12% of the normal number of stomata, did have about a 10% drop in both photosynthesis and in vegetative biomass. So this one is starting to be inhibited by the lack of stomata, but still remarkable that plants can get by with so few stomata. We brought our plants back to Sheffield and grew them in our controlled environment growth facilities to measure water loss. We looked at them with the thermal imaging camera, and again, you can see that the plants with fewer stomata, the low stomata and very low stomatal density lines are slightly warmer because they're not quite as blue as the control line, has more dark blue on it. So this is because the control line is losing more water and has more evaporative cooling. These two lines are slightly warmer because they are losing less water. So we weighed plants and measured how much water they used over a week when they were well watered at about 5 weeks old. And this is to represent how much water each plant on average used. The control plants used, this is a 500 ml shot bottle, just over half a litre of water in a week, whereas the low stomatal density lines only used about 60% of that water over a week. So when they're well watered, they grow with less water. So that's fantastic, but it's only fantastic if they still make the same yield, because making a plant that uses less water is of no use if we have a yield deficit. So we took the plants right through to harvest, and we found that we could find no difference in the yield of these plants. Even the plants that only had 12% of the normal number of stomata produced a similar yield. So in this experiment in our controlled environment facility, all of the genotypes produced about 80 grams of seed per plant. Whether they would do this in the field, I don't know, but under controlled environments, they all had very similar seed yields. So we didn't see a yield deficit. I was a bit worried, though, about them being 0.3 degrees warmer, because under climate change, we're facing warmer climates and more damage to our crops from heat, which can interfere with the reproductive cycle. So I thought that our plants would not be heat tolerant. I was worried that they would be more susceptible to heat stress. Now, normally we grow our rice plants at 30 degrees C. So we did some heat stress experiments, growing them at 35 and 40 degrees centigrade. And to my surprise, all of the genotypes were the same temperature. The plants with lower stomatal density certainly weren't warmer. And I couldn't understand this until I remembered that these plants can still open and shut their stomata. So although they weren't losing as much water when they were well watered, when they needed to cool, they just open up their stomata. So although they have far fewer stomata, they just open them more widely to facilitate that cooling when they need to. So they still have sufficient stomatal capacity to cool the plants, to maintain them. Remember, they're growing at 40 and it's maintaining them at below 30 through cooling. So even with only half or an eighth of the number of stomata, they could still maintain their cooling. So what about drought tolerance? Cereals are most susceptible during their reproductive period. So we imposed a drought just as the plants started to flower, and we stopped watering them for 3 days, which is quite a severe drought for rice plants. And we then re-watered them and took them through to harvest. Now you can see during the drought that the temperature profiles have switched around. Instead of being warmer, our transgenic plants with fewer stomata are now cooler. And this is because the control plants have sensed the drought, they've run out of water and have shut their stomata and are starting to warm up, whereas the plants with fewer stomata have been more conservative in their use of water. They've still got water in the pot, still got water in the plant, and so still have their stomata open, which means they're still transpiring and cooling. And it also means that they're still photosynthesizing, which means that they may be able to maintain a better yield under drought. So we looked at their yields. All of the genotypes had a reduced yield under following the drought treatment. So remember, when they're well watered, they produce about 80 grams per plant. When after this drought treatment, the 3-day reproductive drought, the control plants went down to about 50 grams per plant. The low stomatal density plants fared much better and maintained much better yields, and the very low stomatal density plants couldn't produce a better yield, but did produce a similar yield to the controls. And I think that that's because they didn't recover as well from the drought. So when we rewatered them, because they didn't have as many stomata, they couldn't pull the water back up into the plant as quickly to recover from drought as the other two could. So this tells me that there is perhaps an optimal number of stomata for growth in these conditions, somewhere between normal, 100% of the normal number of stomata, and 12%. So somewhere in between, there is an optimal drought-tolerant number of stomata. And we can actually see how much better this yield was. This is the average amount of grain produced per plant, that the low stomatal density plants produced 27% more grain than the control following this drought condition. And they also had a higher grain weight, suggesting that grain filling was better and would be better quality grain. Now, rice does have a particular problem with water. It uses a lot of water, and it is a big emitter. Rice paddy fields are a big emitter of greenhouse gases. The anaerobic soils of paddy fields encourage the growth of methanogenic bacteria, and methane is about 80 times as potent in global warming as carbon dioxide. Also, the many little pumps driven by electricity and diesel to flood the paddy fields cause a lot of CO2 emissions globally. So paddy rice accounts for 2 to 3% of human-induced greenhouse gas emissions, so has a big impact on climate change. And we really need to be changing the way we grow rice. So rice is grown in paddy fields to suppress the weeds, but also because we don't have available high-yielding, drought-tolerant varieties. So some farmers do use direct-seeded or dry-seeded growth conditions, and the availability of better seed that would allow them to grow with less water, I hope could help encourage the switch towards dry-seeded rice production, and so could help ameliorate these greenhouse gas emissions. What about pathogen tolerances in our crops? Well, the major pathogens of cereals are rusts. Rice doesn't have a rust pathogen, though, so I can show you our wheat and barley results for both brown rust and yellow rust. we saw reduced infection and symptoms of infection by these rusts. And this is work that we did with Jane Thomas at NIAB. So 10 days after inoculation of wheat or barley, you can see that the plants with low stomatal density have fewer pustules. So what happens is rust, the spores germinate, produce hyphae that go in through the stomata. The pathogen grows inside in the interior of the leaf and then bursts back out, producing these pustules. And when there are fewer entry points, fewer stomata, we're seeing far fewer pustules per centimeter. So what I think is happening is that on the control plants, the spores germinate and they grow across looking for a stomate. They find one, they produce an apistorium that allows them to penetrate the stomatal pore and infect the plant. So in this control plant, there are 3 stomatal files, and these are all nice, successful inoculations. In the low stomatal density plants, there are far fewer stomata. There's only 1 file here that of cells that has stomata. It doesn't even have as many stomates in it. This spore has germinated here. It's not found a stomate, and it's carried on growing and carried on growing. And eventually it's just run out of energy and died. It's never found one. So they're far less likely to form a successful infection if there are fewer stomatal entry points. But whether these the level of stomatal reduction is so great as it would inhibit photosynthesis, I don't yet know, because these were quite low, very low stomatal density plants that we did our experiments on. So hopefully I've told you that through making plants with fewer stomata, we could reduce the amount of water loss from a crop, and we could conserve water in the soil, and we could protect yields from drought and perhaps also pathogen stress. So these are really important traits if they work in the field for helping to adapt our plants to future conditions. But the IPCC report that came out this year says that adapting to climate change isn't enough, and that we actually need to start mitigating climate change. That is, to meet our net zero targets or our 1.5 degree warming targets, we actually have to reduce the amount of carbon dioxide equivalents that are released into the atmosphere. So I'm now actually far more interested in the idea of climate mitigation through whether we can really reduce the amount of energy needed for irrigation by all these little pumps that rice uses, whether we could protect yields from a drought and perhaps pathogens and reduce crop losses, which means that we don't have to turn over so much land to crops and produce so many emissions in growing our crops. And then really importantly, if we could grow rice that produces less methane greenhouse gas emissions. But I'm really aware that all our experiments are being done in screenhouses or controlled environments, and we need to get them into a field. But we're using genetically modified plants that are overexpressing a transgene. And there are lots of legal and societal barriers to actually getting these into the field and to farmers and consumers. So we need to think of ways that our crops could actually be used. And I want to tell you about 3 ways we're exploring. Firstly, using the antagonizing peptide, the EPF-like peptide that I told you about, that if we knock it down in Arabidopsis, it makes fewer stomata, just like the EPF overexpresses. So could we use a gene editing strategy that could perhaps be more acceptable? And we're already successfully reduced stomatal density in rice, maize, and beans. And I'm hoping that our maize will go into field trials with the company soon. These are our rice gene-edited plants with low stomatal density growing out in the screenhouse in the Philippines and being tested. If we weigh the pots, again, we can see that 3 gene-edited lines require less water. This is weighing pots throughout their whole life cycle. They require less water to grow. So are more water use efficient? Second way I'd like to consider is through naturally occurring stomatal density traits. There are lots of different cultivars of crops out there that we can look at. The International Rice Research Institute has seed banks, and they've sequenced 10,000 cultivars, the genomes in their 10,000 genome project. And we've looked through those sequences for single nucleotide polymorphisms in our epidermal patterning factor-like gene and found some and accessed the seed from the stores, grown them up. And we can see that rice cultivars that have this SNP in our target gene do indeed have a significant reduction in stomatal density, nowhere near as big as our transgenics, but it shows that variation is there to be identified. And then thirdly, using mutagenized rice collections that are produced non-transgenically in Thailand, where they don't allow any genetically modified experiments. So working with Kasetsart University, we've been looking at their fast neutron bombarded mutants. They have a panel of 200 that are stress tolerant lines, and we've screened those for stomatal size and stomatal density. And just to summarize, in a drought experiment where the control rice reduces its yield by 80%, our mutants that have a low stomatal density only reduce the yield by 53%. So again, they're maintaining yield better following drought in a very similar way to our transgenics. So these are now being bred into rice breeding programs in Thailand. So I want to finish by just saying a little bit about the soybean work that Casper has been leading on. He has a number of projects, but I'm just going to talk about the stomatal density project. We have plants growing in Sheffield, both overexpressing the epidermal patterning factor and gene edited to knock out the epidermal patterning factor, like antagonizing gene, all of which that have we have shown have reduced stomatal density. And we're just doing the experiments now to see if they have any improved crop traits. So far, we think that they might be doing slightly better under drought because they're not senescing as fast as the controls, but we don't have the results yet. But as soon as we do, I'm sure you'll be one of the first to hear because we'll be shouting about it. So I think I'll finish there and just summarize what I've been talking about, that we identified a gene involved in controlling stomatal density in Arabidopsis. And when we overexpressed it, we produced plants with fewer stomata that are drought tolerant. We carried out similar manipulations in cereal crops to produce rice, wheat, and barley with fewer stomata. These plants use less water to grow, and we hope that they will prove to be drought tolerant in the field and will maintain better yields under drought and perhaps pathogen stresses. and also that perhaps they could be used to help address our carbon footprint of agriculture in the future. So I'll finish now, and thank you all very much for listening, and I'd be happy to answer any questions.
Thank you, Julia. That was a wonderful talk. I want to ask you, like, under normal conditions, you don't see like you have an increase in the yield in the low density stomata, but you can keep the same production under the best use of the water. And that is very nice. That's very good. What about if you artificially increase the CO2? Could you increase the yield?
We have grown them at higher CO2 concentrations. The experiments that I showed are at about 450, 500 parts per million, because we can't get our growth rooms any lower than that because we're in the middle of a city by a big roundabout. So nearly all of our experiments are at relatively high CO2. We have tried increasing CO2. I don't think it made a huge difference, actually. On the plants, on the wheat plants, we've had plants that are very severely reduced stomatal density that have a yield deficit, and we've tried growing them at higher CO2 to see if it will alleviate the yield deficit. And it doesn't. And I think that that is because the plants aren't starved for CO2, but because having fewer stomata is actually interfering with anther dehiscence. So the anthers have a row of stomata down the middle that they sort of unzip, and that allows them to release the pollen. And if we take out too many stomata, that doesn't happen. So I think that starts to impair the yields before lack of CO2. Sorry, that's complicated.
Thank you. So one last question. Under heat conditions, you can see that the low density stomata mutants, they open wider. So yeah. Do we know why? So is it because it changed the regulation of the stomata opening and closing, or they are more susceptible?
I think it's because they're experiencing the heat stress. So if they didn't open wider, they would be hotter. I think they're doing it to regulate their temperature. I have to say that we didn't take those plants through to yield. So I don't know whether growing them at high temperature would affect their yield. And it's possible that it would. I don't know the answer to that. All I showed is that as seedlings, they could regulate their temperature. And I'd like to do the experiment taking them all the way through to yield at high temperature and see if it really affects their yield. Because I think it might, I think surely it must have some detriment. But we didn't see it in the experiments we did, and I was very surprised.
Okay, thank you very much, Julie. That was a great presentation. Beautiful results, I should say. We don't have more time for questions. I think there's a couple of them in the Zoom chat. So if you would like to answer them, do that while I'll introduce our next speaker. Next, we will have Dr. Fabian Nogué. And I guess now, Julie, you can turn off your camera.
Okay.
See you. Sorry, I was saying, we have Dr. Fabian Nogué. He is from the Institution Pierre Bourgeon. I hope I said it right, Fabian. He did his master's degree and his PhD in plant biology in the University Paris 6. Then he pursued a postdoctoral scientific position at the molecular biology, physiology, and genetics of Arabidopsis. He moved for this to Australia, to the plant industry division of Xyron in Canberra. And since 1998, he's been group leader on the molecular biology, physiology, and genetics of model plants.
And for this, he combines work with Arabidopsis and Fiscomitrella. He is group leader of the Laboratory of Genetics and Plant Breeding at INRA in Versailles. He's senior scientist. And during the last few years, he has been focusing his attention on DNA repair and recombination mechanisms in two model plants, Fiscomitrella and Arabidopsis, at the genetic, molecular, and biochemical level. In that sense, he is part of the meiosis and recombination group at INRA in Versailles, which has been recognized as one of the leader groups in plant meiosis and recombination in the world. More recently, or together with this, Fabian has also a particular interest in new breeding technologies, especially site-directed nucleases, TALEN, CRISPR, and other plant genome editing tools, and has worked in different projects, including the Genius project. The objectives of this project were to set efficient methods like artificial microRNAs, TALENs, and CRISPR for the targeted regulation or modification of model plants, including Fiscomitrella, which is something I think we will be hearing about right now. And to use these techniques for the generation of new traits that can be imported into important crops. So without anything else, Fabian, thank you very much for accepting our invitation, and you can start now.
Thank you, Jose Luis. It's a great pleasure to be with you guys today. And I would like first to thank you, Jose Luis, and the organizer for this very kind invitation to this very interesting symposium. So, as you said, I will present you the recent result of my team in INRA Versailles in France on the use of CRISPR-Cas9 tools for plant genome editing. And you will see that there will be an aspect on development of the tool and optimization of this tool that we are doing most of the time in a model plant, Fiscomitrium patents, that perhaps some of you guys know already. And I would like also to present you some interesting application of those tools for crops and reinforce what Julie Gray said just before, that this type of tools could be very interesting for breeding and perhaps could tackle the limitation of classical transgenesis. So first, I would like to start with a very brief introduction on the CRISPR-Cas system that most of you know, I suppose. So CRISPR-Cas is an immune system used by bacteria and archaea, not only bacteria. And they use this system to collect sequences from a bacteriophage that will infect them in order to detect and destroy further attack with this same bacteriophage. And in this sense, that's really an immune system that keeps a memory of a previous attack by a phage for the bacteria. The two main actors of this immune system are first the guide RNA that contains a 20 base pairs target that correspond to the bacteriophage sequence, and a Cas9 nuclease that can break double-strand DNA. And once the guide and the Cas9 are associated into the cell, this module will scan the genome to look for the 20 base pair target, and then cut this region of the genome, doing a double-strand break thanks to the two active sites of the Cas9 nuclease. So it's a very efficient system for the bacteria as a defense against bacteriophage. And in 2012, three researchers have shown that it could also be used for biotech application to cut a specific region sequence in the genome. The three researchers are here. So it's Elisabeth Charpentier from MPI Berlin, Jennifer Doudna from Berkeley, and Feng Zhang from the MIT. Emmanuel Charpentier and Jennifer Doudna win the Chemistry Nobel Prize for their work in 2020, showing the importance of this discovery and the usefulness of those tools for researchers. They win this prize mostly for the first application of the CRISPR-Cas9 system, that is to induce a double-strand break in a given locus in the genome. In some cases, the repair of this double-strand break is not perfect, and this can lead to a mutation at the given locus. So the system of targeted mutagenesis is very universal and has been shown to be very useful to produce knockouts of your gene of interest in all six kingdoms of life. And just today, we have shown that this could be used for gene function analysis. And the first presentation was very interesting on this point. And Julie also showed us that this could also be used, this knockout strategy, to produce crops of interest. So as probably all of you know already, the toolbox of CRISPR-Cas has evolved very quickly since 2012. And now we can use inactivated forms of the Cas9 that we called Nikkei's Cas9 that will not produce a double strand break, but that will only produce a single strand break in the targeted locus. And using this type of Cas9, of inactivated Cas9, and fusing them to specific enzyme, like the cytidine deaminase, for example, or the adenosine deaminase, you can have a tool that will allow you to do base editing. And in this case, you will be able to transform a cytidine into into thymine, or with the adenosine deaminase, you will be able to transform an adenine into a guanine. More recently, a new application of the CRISPR-Cas system has been proposed, based also on an inactivated Cas9 that is fused not to a deaminase, but to a reverse transcriptase. This reverse transcriptase that you can see here will be able to make a DNA copy of the desired modification brought by a particular guide RNA that is called a PEG RNA for prime editing guide RNA, and insert the new DNA copy into the targeted sequence. This way, with this type of tool, you can change precisely one or multiple base in a given gene, or do precise insertion or deletion in the same genes. So I would say that for us, for people playing with genome editing, it can be considered the ultimate tool for editing a genome. So now we've got multiple CRISPR-Cas tools in the toolbox. Essentially, those tools have been developed in the animal field for gene therapy. And those tools permit to generate in the genome targeted double-strand break, base substitution, targeted deletions or insertion, and even chromosome rearrangement. So one of the objectives of my group since a few years now is to try to transfer those different tools for editing in the plant genome and try to optimize those tools in order to make them more precise or more efficient. So to start with, I will show you data we obtained with the classical Cas9 for gene knockouts in our favorite model plant, Fiscomitrium patens. So as Professor Reyes is already using this plant as a model in your institute, I suppose that some of you know already Fiscomitrium patens. So there is different reason why Fiscomitrium patens is considered a model plant. First, this moss has a rather short life cycle of 10 to 12 weeks, and this will allow you to do many experiments in a quite a small amount of time. Second, the haploid phase is dominant in its life cycle, meaning that you can directly see the consequence of the knockout of the one copy allele of a given gene. In addition, Pepatens has a very strong regenerative capacity, and you can easily start from a protoplast, for example, and get quickly a very healthy and normal new plant. Finally, the genome of Physcomitrium patens has been sequenced a while ago now, and was in fact the second plant genome that has been sequenced after the one of Arabidopsis. So in order to test the possibility to use CRISPR-Cas for gene knockout in Pepatens, we targeted our favorite reporter gene APT. The role of the APT enzyme in every cell, in every kingdom, is to transform adenine into AMP. And this enzyme is also able to transform 2-fluoroadenine, that is a synthetic compound, into 2-fluoroAMP, which is a very toxic compound for cells. So on this picture, you can see protoplast, regenerating protoplast that can grow on the 2-fluoroadenine just because they don't have any more APT functional in their cells. And the other cells that you can see here are protoplasts that died just because they had a still functional APT gene. So so using this system, we can easily measure the efficiency of editing of our reported gene. And for example, if you co-transform protoplast of Physcomitrium patens with a plasmid expressing the Cas9 and one plasmid expressing a guide RNA that is targeting the APT gene, you can see that some of the protoplast will regenerate and be able to live on 2-fluoroadenine, and the other one will die just because the APT gene has not been knocked out in those cells. So if you have a control with only the Cas9 that has been transformed into the protoplast, you can see that all the cell will die. So we have here an easy way to measure the efficiency of knockout of the APT gene. And in these conditions, in routine, we can observe that in average, 52% of the transfected protoplasts are mutated by the CRISPR-Cas9 system. So we could demonstrate that the CRISPR-Cas9 is very efficient in Pepatens, and in addition, we could show that it can target multiple genes at the same time using a mix of guide RNA. And as shown on this slide, quintuple mutants could be obtained using a CRISPR-Cas9 strategy just by transforming protoplasts with the Cas9 and the different guide RNA that will target the different genes on the different chromosomes of Pepatens. And in those conditions, we could observe that 10% of the mutants correspond to the quintuple mutants, and we could find all the different combination of mutation. So this is the type of strategy that we use in collaboration with José Luis Reyes to knock out the metal-13 and metal-14 genes in Physcomitrium, the genes that are involved in its N-cis methyladenosine modification of mRNA in this loss. So now I would like to show you an example of multiplex gene editing in gene knockout in a crop. And first, I would like to say a few words on Camelina that could be considered by some people as the missing link for researcher between the model plant Arabidopsis and canola. Indeed, Camelina can be grown in a greenhouse, even if it's a bit taller than Arabidopsis. It's still smaller than rapeseed or canola. And the gene sequence between Camelina and Arabidopsis are very conserved. You can have as high as more than 90% of conservation for different genes. And the genome of Camelina is available now. And last but not least, this plant can be transformed by floral deepening like we can do with Arabidopsis. So we can easily produce mutants, and in many cases, when we develop CRISPR-Cas9 strategies, we can use the same guide RNA that we use in Arabidopsis into Camelina. So Camelina can be also of interest for agriculture. It was widely grown in Europe and Russia until the 40s for the same purpose as canola oil and feed, with an oil that is very rich in omega-3, which is a quite healthy compound. Camelina is now also used in cosmetics and for biofuel. And the major advantage of Camelina is that it needs only very few inputs to grow. And this is the reason why there is a new interest for this crop in Canada or Europe, for example, and especially at INRA, we are quite interested by this crop. So one possible use of Camelina could be double cropping. So in Europe, there is 3 to 4 months between the harvest. For example, for barley, you can have 3 to 4 months of what is called off-season, where you will have nothing in the field before the sowing for the winter barley, for example, that will be done in October. So between July and October, you've got a field that is naked, where there is nothing growing. And one idea would be to use Camelina as a possible crop to be grown during this very short period. So for that, we would need short, early flowering Camelina, and we try to look for that in the breeding pool of Camelina, testing different more than 200 varieties of Camelina, and we could not find a variety that could be early enough to be able to grow it and collect seeds between this rather short period. So that's why we wanted to try another strategy, and we decided to generate an early flowering Camelina using the CRISPR-Cas strategy. So we call this the targeted induced genetic variation strategy, and the principle is this one. We choose a group of genes involved in a given pathway. In our case, this is flowering, and we design guide RNAs that will target all those different genes. Then we transform a wild-type Camelina with the different guide RNAs that will target those group of genes, and we will just screen directly for early flowering Camelina in a bulk of transformed plants without looking at the genetic status of those plants. So the first screen is on the trait and not on the efficiency of editing. So this is what so you can see on this slide, sorry, the pathway for flowering that has been established in Arabidopsis. You can see that it's a very complicated pathway, and there is 5 major genes that have been shown to inhibit flowering time, and this is the type of genes that we would like to knock out. And this is LHP1, FLC, SVP, TFL1, and ELF3. So we focus on those 5 genes, and we design 5 different couples of guide RNA, with each couple targeting 2 regions in the 5 candidate genes. Because Camelina is an hexaploid, it contains each time 3 copies of a given gene. So we had to select guide RNA that would be able to target the 3 copies for each of our 5 genes, and we produce 2 guide RNA per genes. So this means that we had 10 guide RNAs targeting 5 genes that are all in 3 copies. So we grouped those 10 guide RNAs on a binary vector with the Cas9 and with a visual marker, the DS-RED, that will allow us to select for transformant, but also later in the process of screening for plants that would segregate the Cas9 construct, because what we would like at the end of the story is an edited plant, but with no more Cas9 in it. So the primary transformant using this construct have been screened in greenhouse for time of flowering, and different lines could be selected as early flowering. These lines were selected until the T5 generation in presence or absence of the Cas9 construct, and early flowering phenotypes was checked and measured at each generation. And you can see here, for example, an example at generation T3 for a given line on that flower early compared to the control plants. In total, we could select 15 early flowering lines with one line flowering 9 to 10 days before the control line. So the best variety that we could find in the breeding pool used by the breeder at the moment was only 4 days before the flowering before the wild type. So using this strategy, we could find a plant that could flower a lot earlier than what was disponible in the breeding pool. We then established the genotype of the most interesting line. It's just an example. I don't want you to look at each line, but just to show you the complexity that of the combination that we could obtain in the 15 different genes that were targeted. All those lines are now fixed and do not contain the Cas9 anymore. For sure, such a complex genetic combination would have been very difficult to obtain by classical breeding. Of course, the next step now, but this is a bit difficult in France at the moment, would be to measure the flowering time of these different lines in field conditions, because I remind you that all the screening and the checking of the phenotype has been done in greenhouse at the moment, and we really would like to see how correlated is the flowering time in the greenhouse compared to real field conditions. So here we were limited by the gene editing tools we use, as we could induce mostly knockouts of the targeted genes. So it has been quite successful, but obviously we could even increase the time, the earliness of flowering by targeting, for example, genes that are inducers of flowering. And we would like to be able to modify the expression, for example, of this type of genes, or be able to modify specific amino acids of some of those genes that are involved in the induction of flowering. And this type of strategy could be very complementary to the first one, and perhaps we could accumulate some of those mutations in the future. So we need more tools, as I told you, for gene editing. And this is why I would like now to talk about a more precise way to modify genes that is the base editing strategy. So to test the possibility to use base editing in Fiscomitrium patents, we targeted the APT reporter gene I presented you before with a cytidine deaminase fused to a nickase Cas9. We use a nickase Cas9 here because you need, as I told you before, a single strand break to fix the mutation induced by the cytidine deaminase enzyme. We use this type of enzyme to target the APT gene with targeting the guide RNA2 and targeting the cytosine that are present in this guide RNA. We did the same thing, but with the adenine deaminase and targeting another guide RNA that was rich in adenine this time. And we used this adenine base editor that is composed of an adenine deaminase TAD-A duplicate that is an enzyme that is present in E. coli and still fused to Cas9 nickase. So first we compare the respective mutagenic activities of the two base editor compared to the classical Cas9 for the two different targets. For this, we co-transform guide RNA or guide RNA1 or guide RNA2 with the Cas9 or with the respective base editor and selected for APT mutants. You can see on this slide that the mutation efficiency using the base editor is decreased compared to the active Cas9, but is still high enough to be used to produce variants for a given gene in Fiscometrium patents. And in this case, we could observe that efficiency was 21% for the cytidine base editor and 14% for the adenine base editor. In order to characterize the nature of the mutations that were induced by the base editors, we sequenced the APT gene in some mutants, and you can see here that majority of mutation, 89%, correspond to clean base editing events for the cytidine base editor with modification of a C into a T, a G, or A. And for ABE, we can see that the situation is even more simple, as 100% of the editing correspond to an editing of an A to a G. I would like now to show you an example of an application of this cytidine base editor we developed and optimized in Fiscometrium patents for gene function analysis in tomato. So this work has been done in collaboration with Jean-Luc Gallois from INRAE of Avignon in France, and it concerned the role of the translation initiation factor called EF4E in the resistance to potiviruses. So potiviruses, like the PVY virus, use the EF4E protein to establish its cycle of infection, and can be and this type of virus can be very detrimental for Solanaceae plants like tomato, potato, or pepper. So resistant variety to potivirus exists, in particular in pepper, and it has been shown that two regions of the EF4E protein are involved in the resistance to viruses, the region 1 and the region 2 on this protein folding prediction. In tomato, the wild tomato Solanum abrocates contains an allele of EF4E named POT1 that gives resistance to a broad spectrum of potiviruses and strain. The POT1 allele contains a number of amino acid difference compared to the cultivated tomatoes, with 5 mutations located in the region 1 and 2 of the protein. The POT1 allele is very interesting in terms of resistance, but is very difficult to introgress into cultivated tomatoes due to the difficulty of crossing between this wild tomato and the cultivated ones. Nevertheless, a near isogenic line has been produced, but it took many years to establish it. And it has been done only in one specific domesticated variety of tomato. So this is the reason why we decided to have a gene editing approach to try to mimic the POT1 allele in more than one cultivated tomato. And for this, we first targeted region 1 of the EF4E gene and decided to target the proline in position 69 that was already shown to be potentially involved in resistance to PVY virus in pepper. So for this purpose, we use first a gene knock-in strategy through CRISPR-Cas9 with 2 different steps. We deliver first through particle gun a recombinant matrix containing a proline to threonine change of amino acid number 69 that integrated into the EF4E gene thanks to the induction of a double-strand break in the targeted homologous region of EF4E. In a second step, we eliminated through CRISPR-Cas9 the kanamycin-resistant cassette that allowed us to identify the first integration event that in flowering plants is always a very rare event. This way, we obtain a new allele of EF4E, where the proline of the region 1, the proline 69 of the region 1, was modified into a threonine. The strategy looks very straightforward on the slide, but it took us a very long time, more than 2 years, to obtain a clean version of the new allele. So for this reason, we choose another strategy, base editing, to modify the second region of the EF4E gene. For this modification of region 2, we delivered into wild-type plants or plants already containing the P69 modification, the citidine base editor targeting the sequence present in region 2 that you can see on this slide. Using this strategy, we could produce in less than 1 year a new variant of the EF4E gene containing 2 amino acid changes in the region. The glycine of position 110 change into a glutamic acid, and the alanine of position 111 change into a threonine. We then tested the resistance to the PVY virus of these different alleles. And you can see on this slide a DAS ELISA quantification of PVY accumulation in plants after inoculation. So you can see that we could produce lines that are a lot more resistant to the PVY virus compared to the wild type and are close to the POT1 allele that is present in the wild tomato. Interestingly, you can see that mutation in one of the region or the other is not sufficient to gain a resistance to PVY. And you can see on this picture that in vivo, in planta, you can also see the multiplication of the virus in the wild type or in a single mutant compared to the absence of infection in the different alleles resistant to PVY. We could observe that those resistant alleles were resistant to more than one strain of the PVY and also to other potiviruses like the POT1 allele. And again, that the single mutation was not sufficient in region 1 or in region 2 to produce a resistance. So now that we have been able to produce those plants, we would like to be able to decipher the different amino acids that are important for the resistance and compare, for example, the modification that are in POT1 compared to the resistant line that we have produced that contain less mutations. And for that, we would need a better tools than base editing, and we would like to be able to use prime editing. So now I will really briefly talk about this new CRISPR-Cas tool that we have tested in Fiscometrium patents and give you an idea on the potential of this tool for gene function analysis and breeding, but also, of course, of its limitations. So this prime editing is composed of, as I said before, of a prime editor that is a guide RNA to which has been added a primer binding site and an edited RNA that will serve as a matrix for the modification that you want to induce in your targeted plants. And it's composed of a reverse transcriptase associated to the Cas9, and the reverse transcriptase will be able to transform the edited RNA into DNA, and this DNA can be integrated in the targeted locus thanks to the FliPase system. So we tested this strategy, these techniques on Fiscometrium patents, of course, and on the APT gene, and we produced different PEG RNA that target the APT gene with different type of modifications. You can see here the efficiency of those different PEG RNA that is rather low. And in fact, even for the best PEG guide RNA that we could find targeting the APT gene, the efficiency of mutagenesis, the mutagenesis rate is quite low compared to the efficiency with the native Cas9 that is 8.4%. And here we've got 0.4% of efficiency. we analyzed the nature of the mutation induced by the prime editing, and the blue part of the histogram here corresponds to mutants where the predicted modification could be found. So we can conclude from this data that even if mutation rate is low, editing is rather precise, and prime editing can even reach 100% of clean editing, sorry. Nevertheless, as I told you before, the editing efficiency is probably too low for us to envisage to use prime editing in species like tomato or potato. So for this reason, we try to improve the prime editing efficiency. And for that, we use we base our work on a study done in animal cells showing that adding in 3' of the PEG RNA a specific RNA structure could improve the efficiency of prime editing. So we did that in Fiscometrium patents, and we could see that at least for each PEG RNA, the increase in efficiency was 4-fold and could even reach more than 130-fold for a given PEG RNA. So just adding those motifs at the end of the PEG RNA stabilize the PEG RNA and increase a lot the efficiency of editing. To go a step further, we tried to stabilize our PEG RNA with a TLS-like structure coming from a plant virus, and we could show that this sequence could, like EvoQ1 or MPIC-NOT, those specific structure, could increase the PEG RNA efficiency for all the different PEG RNA we tried. So I will not go too much into the details, but I just wanted to show you that integrating also extra mutation in the RNA matrix that is used for the modification can also increase the efficiency of editing, as you can see here. So combining those different strategies, I think, can lead us to a more efficient prime editing ratio. And you can, in this condition, you can see that efficiency of editing is only 2.5 less efficient than the classical Cas9 strategy. So we really reach numbers that should allow us to use this type of strategy in crop plants. So just a few conclusions. Gene knockout and base editing are efficient in Fiscometrium patents, precise. Off-target of the different tools that I've shown you is low if detectable, and you can see our last paper on this subject if you are interested by off-targeting. These CRISPR-Cas tools can be used to produce new varieties in crops, and I think that Julie showed us in our previous presentation, and I hope I convinced you that those tools can also be useful for camelina or for tomato, for example. For me, prime editing is very promising, and PEG RNA stabilization is clearly crucial to increase the efficiency of prime editing. And there is probably still some space for improvement, like playing, for example, with the reverse transcriptase, because at the moment we all use the same reverse transcriptase, and perhaps we should try other ones. We have already started a test of those new PEG RNA strategy on potato and tomato, and we hope to be able to show you results soon on the efficiency of prime editing in those two Solanacea species. I would like to thank you again for your kind invitation and attention. And I would like now to thank the colleague of my group who participated to this work, especially Anouchka Guyon and Pierre-François Perroux, and also Jean-Luc Gallois and Jean-Denis Faure, with whom we are collaborating on the tomato and camelina projects. And I would like again to thank José Luis for the invitation, and thank you again for your attention. Thank you.
Thank you, Fabian, for that. Your talk. It's very impressive the amount of work you've done. I don't see hands up, so I may want to do a first question. And that is, I'm looking at you, but you may not be looking at me. So I'm going to move around here, I guess. Anyways, so in this last concept about the RNA and the folding or the stability, Are you thinking of doing experiments like in vitro selection of the most stable structure, or how is that done?
Yes, so you are a specialist of RNA, so probably I will have to discuss with you this aspect. But yes, I think that clearly a key point for prime editing efficiency is the stability of the guide RNA that you will use. So you can try to use. We try to use some prediction tools for the stability of our guide RNA, but clearly those tools have their limitations. It's not exactly what happened into the cell. So at the moment, yes, we are trying to think about a strategy that could give us an idea of the stability of a candidate peg-RNA. And in this case, we should be able first to screen for the best peg-RNA and then do the experiment in the plant. For Fiscometrium patents, you know how easy is the transformation of Fisco. But for plants like tomato or for potato, you will really need this step of selection of your peg-RNA before starting the experiment of transformation.
Okay, thanks. We have another question here.
Thank you for your nice talk. I'm just wondering whether when you say that the frequency of off targets is low, that happens in the different varieties or species that you have tested?
That's a good question. That's a very good question. Yeah. So the only study that we have done is with CRISPR-Cas9 and with TALEN, which is another tool that can be used for double-strand break induction. And we have only done that in Fiscometrium patents. So using those two different tools, we have been able to show that the nuclease by themselves does not lead to mutations. But intriguingly, the process of transformation of the protoplast of Fiscometrium was mutagenic. Just a little bit mutagenic, but it is mutagenic. And in fact, when you transform protoplast, you use PEG transformation, PEG transformation. And it has already been shown a long time ago that in yeast, this molecule could be mutagenic just because it causes a shock into the cell and into the nucleus that can lead to some mutations. So we could see some mutation in our transformed plants, but only due to the PEG transformation and not to the CRISPR-Cas9 itself. So we have shown that in Fiscometrium. And of course, now we would like to show that also, or to look at that in tomato especially. And we have started experimenting in this direction.
Thanks. Thank you, Fabian. We'll close with two questions that are in Zoom. I don't know if you can see the chat. One is from Armando Bravo. I'll read that one first, and then Patricia has her hand raised. And then we'll stop for a coffee break. It says, great talk. Did you check the efficiency using different ways of expressing the guides? For example, the RNA system to produce a single polycistronic sequence with multiple guides.
Yeah, so that's a very interesting question. So as I have shown, the stability of the PEG-RNA is very important. So most probably the level of expression of this PEG-RNA is also probably very important. So at the moment, we haven't tried a polycistronic system, just because in Fiscometrium patents, it's very easy to transform with different PEG-RNA that are on different plasmid. It's really not a problem to do so. But in Arabidopsis, in tomato, and in potato, we are envisaging two different strategies. The classical strategy with different PU6 promoter that permit the production of the guide RNA, and also a polycistronic strategy with a classical 35S promoter, for example, and try to see if this could make a difference. For classical guide RNA, this does not make a huge difference, but perhaps for PEG-RNA, where stability is clearly more important than for classical guide RNA, perhaps this could be very useful to use this type of strategy.
Okay, if you could very quickly answer the other question in the chat from Patricia. I saw that you use wild tomato variety. Is this due to the regeneration facility?
So yeah, it's true that wild tomato can be very useful on this point of view. But in this case, we use wild tomato only for the purpose of the resistance to the PVY virus. And perhaps I was misleading, because when I say wild type in the table looking at the resistance to the PVY, for me it was wild type of a domesticated cultivar, okay, of a cultivated tomato.
Okay, thank you very much, Fabian.
Thanks a lot.
It was a pleasure seeing you again.
Thank you.
Well, now we will have, I guess, a 10-minute coffee break for the ones who are here seated at the auditorium, and we'll see you in a bit. Bueno, vamos a reanudar la sesión. Todavía tenemos dos pláticas. Perdón. We're going to reinitiate, to return from the break. And to present the next speaker, I'll let Dr. Omar Pantoja do it.
Gracias. Buenos días. Good morning. And, I would like to introduce the next speaker, Armando Bravo, who is he spent 2 periods at the institute. First as an undergrad, with doing his thesis with Dr. Elena Porta, and then he went to do his PhD to University of Oxford with Jean Landale, working on comparative study of the role of transcription factors in chloroplast development and evolution between algae, bryophytes, and angiosperms. From there, he came back to Mexico and spent some time in my lab, where he was a bit unlucky because he couldn't get good results. But anyway, from there, he went on to get a postdoc with the Dr. Maria Harrison at the Boyce Thompson Institute, where he did a very good work with mycorrhizals. And from there, he is now a principal investigator at the Donald Danforth Plant Science Center, working on the functional study of arbuscular mycorrhizal symbiosis using an evolutionary framework. Thank you very much, Armando. And
can you hear me? So, hi, let me see. Can you hear me?
Yes, you're okay.
Okay, great. Thank you. Well, thank you very much, Omar, for the introduction, and thank you very much for the invitation. It's like really very exciting for me to be presenting because, as Omar said, I've been at IBT a couple of times in very important stages of my career. So actually, reflecting about the theme of the symposium that is titled Our Roots, Branches, and Fruits, Pepe already mentioned that I'm considered, I guess, part of the fruit part of the analogy since I was a student there. However, actually, from my point of view, I consider myself more of like the roots part of this analogy because not only I work with roots, but also the roots of my knowledge and my passion about plant science and in general my career are there at IBT. So first, I was an undergrad working with Elena Porta in Mario Rocha's lab. That's where I learned the basis of plant molecular biology. So thank you very much, Elena. And also a few years later, when I finished my PhD, I went back to work with Omar Pantoja as a postdoc. And there I learned a lot about plant physiology and ion transporters, which boosted my interest in plant nutrition, which eventually led to me moving into mycorrhizal symbiosis research, which is what I will talk to you about now today. So why study mycorrhizal symbiosis? So we all know that plants are limited by the amount of nutrients that are available in the soil and that nutrient-poor soils are not good for agriculture. So that's why we use fertilizers. But we also know that chemical fertilizers are not good for agriculture. And the use of this chemical fertilizer is not a sustainable practice because the resources are limited and it contaminates our planet. So one example is phosphorus, that we need to mine as phosphate rock from just a handful of places in the world. And this is a finite resource that has been estimated to only last for a couple hundred years more, which is not that much. In addition, from all the fertilizer that we apply to the plants, only a fraction of that is going to be used by the plants themselves. So for the most part, we use an excess of this fertilizer, and a huge amount of these will be lost into runoff that will eventually reach different rivers and lakes and even the sea. And when it reaches these bodies of water, like lakes, that it will be used by other organisms, such as algae. And this will cause a huge contamination problem that is even harmful for human health. So to find a solution to this problem, what we need to do is to look back at how nature deals with this. And the problem of nutrient scarcity is not new for plants, we know, since the beginning of their evolution, particularly during the colonization of land, plants have been forced to increase their resilience to the harsh conditions that they were faced during this movement to land. And through time, plants have developed multiple strategies to cope with these conditions. One of such strategies eventually gave rise to the formation of roots. But even way before roots, plants actually established interactions with microbes that would help them endure these harsh conditions. And one of the most important of such associations was with mycorrhizal fungi. So fossil evidence suggests that the earliest land plants were already forming associations with fungi that look very much like modern-day arbuscular mycorrhizas. So molecular data also supports the notion that mycorrhizal symbiosis originated really very early at the beginning of land-plant evolution. And these interactions still persist in the great majority of the lineages that we see today, maybe with just the exception of mosses, but all major other lineages still form associations with mycorrhizal fungi. And this goes from bryophytes to ferns and to flowering plants. And this makes this association, this mutually beneficial association, the most widespread and ancient relationship that we know. Now, arbuscular mycorrhizal symbiosis, which is one of the main types of mycorrhizal symbiosis that exists, and which is abbreviated as AM symbiosis, it's the mutually beneficial association between the great majority of land plants, so around 80% of all land plant species can form this association, with only one group of fungi, which we call arbuscular mycorrhizal fungi, and belongs to the group of Glomeromycotina. Now, in this relationship, plants get multiple benefits, which you can see there listed, but maybe the most notable of them is the enhanced mineral nutrition. And during this association, plants will provide the fungi with carbon that they produce during photosynthesis, and in exchange, they will get mineral nutrients, mainly phosphorus and nitrogen, which are more accessible to the fungus. And this is of great importance, not just for agriculture due to the increased plant nutrition, but also to ecosystems because of the cycling of these nutrients. And by studying mycorrhizal symbiosis, we can leverage our knowledge to better manage this interaction. And we can improve then plant nutrition and health and still protect the environment. Now, arbuscular mycorrhizal symbiosis gets its name for the highly branched structure that is called the arbuscule that you can see here. So this is a very beautiful, highly branched hypha that grows inside inner cortical cell layers, inner cortical cells in the root. Now, so that you can have a better idea of how that looks, this is an image of a root that has been cleared so that we can see through, and has been colonized by the mycorrhizal fungus and has been stained. So whatever you see in green there is the fluorescent dye that binds to the fungal cell wall. So everything that you see is fungus. And each of these little blocks that you can see here, let me see if I can see my pointer. I cannot see it, but anyway, like these little blocks that you can see here are the cells that are completely filled with the hyphae, the fungus that has branched completely. And that would look something like this. This is a close-up of a cell that is completely filled with this fungus that is branching and growing inside of the cell. In this case, in this image in the middle, what you see is actually a plant protein that is localized in a plant membrane. So this is a protein fused with GFP that is localized into this membrane, which is called the periarbuscular membrane, that completely surrounds the fungus. So in this cartoon on the right, you can see the membrane that completely surrounds the fungus as it is branching. So there's an analogy of this that you can imagine a hand and a glove. So the hand would be the fungus penetrating the cell and branching, and the glove would be this periarbuscular membrane that is a continuation of the plasma membrane. And this arbuscule and this membrane is where most of the transporters are localized, and the arbuscule is the site of nutrient exchange. So it's very important for the symbiosis. And in order to better manage this interaction, we first need to understand how this is controlled, the molecular mechanisms that regulate this. And while our knowledge has increased in the last few years, we're still kind of far from getting the whole picture. And in this cartoon, I just want to show you what was known when we started this project a few years back. This cartoon represents a plant cell that has been penetrated by a fungal hyphae, and that is always surrounded by a periarbuscular membrane. And these are the mechanisms, these are the genes that had been identified at the time that were involved in mycorrhizal symbiosis. This was again a few years back. So at the time when we were trying to figure out how to study this, we're trying to identify new genes that were involved in this symbiosis. So the main question was, how can we identify new genes? And traditionally, there's had been two types of genetic screens that, in order to look for these new genes, there are forward or reverse genetic screens, and each one has their own problems. In the case of forward genetics, it's technically difficult because of the nature of this association below ground. In the case of the reverse genetic screens, it's because they're based on expression data we're talking about thousands of genes that are induced during symbiosis, and it's difficult to pinpoint which are the important ones. So what we did was to develop this strategy that is based on an evolutionary hypothesis, and which is called phylogenomics, that we can use to identify new genes. And that's what I'm going to explain to you briefly here in this slide, which represents a cladogram of 48 plant species whose genomes have been fully sequenced. They include both monocots and dicots. Of course, they are biased towards what was sequenced at the time. This was like, again, a few years back. Now the number of species has grown dramatically. But back then, like these species, which represented what we knew, they are color-coded based on their ability to form symbiosis. So in black are those species that form the symbiosis, and in red, those plants that do not form the symbiosis. And the important thing is that, based on what I mentioned earlier, we hypothesize there is a single origin for symbiosis, which means that all the lineages that don't form symbiosis in extant plant species is because they lost this ability to form the symbiosis. And because now we have this genomic information, we have a whole repertoire of all the genes. Now we can make comparisons and then identify those genes that are important and present only in plants that form symbiosis. So kind of a way to see this in this example, the subset, is to imagine a small set of species that form symbiosis in black, or in Arabidopsis thaliana, notably, that doesn't form symbiosis in red, and they all have a common ancestor. And this common ancestor, you can imagine that it had all the genes that were required for symbiosis. However, through their evolution, some of these genes were lost for some reason. And therefore, now the extant plant species that form the symbiosis have all of the genes, whereas plant species like Arabidopsis that do not form the symbiosis, they have lost some of those genes. And all of those that have been lost, so the ones in gray in this case, are the ones that we call the genes conserved for mycorrhizal symbiosis. And we hypothesize that because they have been conserved only in those plants that form symbiosis, therefore they should be required for the symbiosis and therefore important. So how do we compare this? Is by analyzing the genes that are common for different groups of plants. So in this case, monocots and dicot plants. And then we see what is in common between them. And then from everything that is common between them, we remove everything that is present in plants that do not form the symbiosis. What we end up with is with that circle, well, that area in the middle, which is only common among plants that form symbiosis. And that's what we call genes conserved for mycorrhizal symbiosis. And effectively, the way we did this, because we were not the only group that tried this, but I think we were the most successful in finding these genes, is because we did this by doing phylogenies. So getting to the basics of creating phylogenetic trees, which are very important. And we did automatically phylogenies for all the genes in those different plant species. And then using an automatic pipeline, we have detected which ones of those lineages, which one of those clades only contain species that form symbiosis. And those are the important ones, the ones that we care for, that form the group of mycorrhizal conserved genes. Now, how many genes each plant has depends on the species because of duplications and so on. In the case of Medicago truncatula, which is this legume model system that we use for mycorrhizal symbiosis, and which has around 44,000 plus predicted coding genes, there's only 138 genes that we identified as concerned for mycorrhizal symbiosis, which is a workable number, so not too big, not too small. And to figure out if we were kind of on the right track, one thing that we did was to compare how many of those genes had already been identified. And previously, at the time when we published this, there were already 36 genes that had been identified important for mycorrhizal symbiosis, out of which 14, so just a little bit less than half, belonged to this category of concern for symbiosis. And fast forward a few years, and nowadays, the number of genes has increased, and so has increased the number of genes in the list of conserved genes, and like around also a little bit less than half of the genes that have been described, which tells us that we're kind of on the right track. So many of these genes that we identify are really important for the symbiosis. And another thing that we learned is that not all of the genes belong to this category, or at least we cannot detect them with the methodology that we used. And there's reasons for doing that, because we know that several genes have other functions other than mycorrhizal symbiosis, so have shared functions, and therefore could be present in plants that do not form the symbiosis. But the most important thing is that we can detect new genes, which is what we wanted. And to figure out also if we were on the right track at the time, what we did was to compare the expression data that we had on genes. So in this pie chart, what you see is genes from a microarray data set that included genes expressed in roots. And the green part of the green fraction of this pie chart represents that the genes that are upregulated during symbiosis, which we know that's what happens in genes that are important for the symbiosis. It's around 50% of the genes, and still we're talking about thousands of genes. However, if we take a look at the genes conserved for AM symbiosis, the great majority, like around 85% of the genes, were induced during symbiosis, which tells us that our methodology was quite accurate in identifying these genes. However, the ultimate proof, of course, is a functional analysis. So what we did is trying to look for the function of these genes using mutants. So we searched a collection of mutant plants in Medicago and identify 6 different lines which were mutated in 6 different genes that were inserted by a retrotransposon, creating a truncated protein and a non-functional protein. And what we did was to evaluate their ability to form the symbiosis and to see how much of the root was colonized, and this is a proxy of how successful the symbiosis is. What you can see in these graphs is that each pairwise comparison between the gray bars that represent wild-type plants and the green bars that represent the mutants, we always saw a reduction in the colonization, which means that these plants are affected, impaired in the formation of symbiosis, and therefore these genes are important for this symbiosis. So confirming that our way of identifying these genes was fairly successful. So we published this a few years back, and what have we learned from studying these AM conserved genes? So what I'm going to tell you is a couple of stories that what we have learned. The first one is about a phosphate transporter story, and the thing that we learned, this kind of a spoiler alert, is that many of these AM conserved genes have major roles during mycorrhizal symbiosis, and I'm going to explain why. The other story that I'm going to present you is about lipid biosynthesis, and what we learned is that plants have readapted many of the basic pathways that they already existed. So not to reinvent the wheel, they just modify some of the key regulators to fine-tune these larger pathways. And that's how we also identify lipids as being so important for mycorrhizal symbiosis. So for my first story about phosphate transporters, we knew for a while that there was this phosphate transporter called PT4 that was identified in Medicago truncatula, in the legume plant, and that when plants lack this transporter, they cannot form a successful symbiosis. So the cartoons that represent a wild-type arbuscule will show like a highly branched arbuscule, whereas plants that are mutated in this PT4 transporter have what we call a collapsed arbuscule that doesn't branch nicely, and there's not much exchange of phosphorus. Now, this gene belongs to this category of AM conserved genes. And the interesting thing is that when we look at other plant species, we see still the same AM concern genes, but other stories, other genes that are also induced during symbiosis, which is not the same case in Medicago. So we took a look at the grass Brachypodium distachyon, which is also another model system. And we found that what we saw actually is that there's multiple of these phosphate transporters that are being induced. And that's what you can see in this image on the left. So the question was, is there a different way of regulating this phosphorus exchange between the plant and the fungus in this plant? Is different to the other plants? Maybe because there's multiple transporters now, they have different overlapping roles, maybe. So to try to answer that question and to try to look at the importance of the different genes, we took an approach of generating mutants. So we use this CRISPR-Cas9 that Fabienne already explained very nicely, and in which we generated mutants for all these 4 different genes that were induced during symbiosis. What we were aiming to do was generating single, double, and even quadruple mutants for all of them, because our original idea was, since there are so many genes that are being induced, maybe we need to knock them all out in order to see an important reduction in colonization. As a side note, also, we were using a different system, but it's very effective in knocking out genes in Brachypodium distachyon, because even in the second generation, in the first generation, actually, we already had homozygote mutants for double mutants. And it only took 2 generations to get homozygote quadruple mutants of these Brachypodium plants. So what we saw after generating these mutants was taking first a look at the morphology of arbuscules. This is how wild-type arbuscule looks like highly branched arbuscules, filling most of the volume of the cell. Whereas when we see the mutants, we see that they are impaired in the symbiosis. Interestingly, and I guess a bit well, we expected a little bit that. I guess that the PT7 gene that is homologous to the known PT4 gene forms these collapsed arbuscules. And what we saw is that plants in Brachypodium only require that gene to be knocked out to have this collapsed arbuscule mutant phenotype, which tells us that this is the main transporter involved in the development of arbuscules. In contrast, for example, the double mutant of PT12 and PT13 forms completely wild-type, highly branched arbuscules. And when we look at the growth of the plants, we see basically the same thing. So in these images, you can see the plants grown with and without fungus next to each other. And you can see that the plants that have the fungus, in the case of the wild type, grow much better. So that's the effect of growing plants in low phosphorus conditions and having the fungus help them to acquire these nutrients. And as you can see, in all of the cases in which there are mutants with PT7, there is not such increase in the biomass, which was measured and quantified on the right graph that you see there. And with the exception again of the PT12-13, because there's a functional PT7 gene in there. So kind of the conclusion is, in order to have this beneficial effect in the growth of the plant, we need the plants need the PT7 gene. However, we were thinking, but maybe, okay, even if it's not for the growth of the plant, maybe just the exchange of the nutrient, maybe to a lower level, you would see something. So we were trying to see whether there were differential roles in the different phosphate transporters in the exchange of the nutrients. So what we did is using this system in which we have the plants grown in these pots, that we have a little kind of vial that has some phosphorus, radioactive phosphorus, that is covered by a mesh, so that is only accessible by the fungus, and the roots cannot penetrate that. So everything that gets the radioactive phosphorus is going to be transported by the fungus into the plant. So then we were trying to grow the plants as equally as possible so that there was not a difference in biomass. That's why what you can see in the graphs on the right. But also we wanted to have some functional symbiosis going on, because as I showed before, like the arbuscules are collapsing in all the cases that there is a PT7 mutant. So we kind of play with the conditions, trying to figure out a point in which there was still some symbiosis going on. And then we measured the radioactivity and how much of this radioactive phosphorus was taken up by the fungus into the plant. And what we saw is that in every single case in which there is a PT7 mutant, so the PT7 is not functional anymore, there is no transfer of the phosphorus anymore. So which tells us that it's kind of a very drastic effect of the PT7. The function is pretty much only PT7 transferring the phosphorus through the mycorrhizal symbiosis. And the function of the other post-phosphate transporters might be different in other different things. So now we know that this dominant role in genes conserved for symbiosis is not the rule for all the genes that we have identified. And as we keep studying the list of AM conserved genes, we keep finding new interesting things. And this pie chart actually summarizes the knowledge that we have generated studying these genes, including both published and non-published data. And we've found from low colonization phenotypes to even higher colonization, so negative regulators of symbiosis, different interesting mutant phenotypes that we have seen. And one of the ones that I want to talk about is one of the original ones that we identified, which is called FAT-M, which is a gene that is related to lipids. This gene, the mutant for this gene produces these collapsed arbuscules that you can see in this image. And one thing that we knew from other studies is that there were other lipid-related genes that had a very similar mutant phenotypes. So we were trying to figure out what's the importance of the lipids in mycorrhizal symbiosis. And so in the next slide, what I'm going to do is going to summarize a bit of how the lipid pathway works in plants in general. So we know that in de novo fatty acid synthesis takes place in plastids, and after that the lipids move into the ER, in which what is known as the Kennedy pathway will modify these lipids into different membrane and storage lipids. However, there is a key enzyme called GPAT, glycerol 3-phosphate acetyltransferase, that will divert the production or modify these lipids to produce a different type of lipids, which are called monoacyl glycerols, which are cutin and suberin precursors. So in different contexts of different cells, they work in different ways. So in the case of epidermal cells in the leaf, for example, they would produce these monoacyl glycerols, which are going to be transported by ABC transporters out of the cell, and they will form the plant cuticle. So that's their function. However, in the context of an inner cortical cell in the root that is being colonized by mycorrhizal fungi, what happens is that there's going to be the same, the same group of enzymes, but it's a specific gene that will produce the same monoacyl glycerols, be transported into the fungus so that the fungus can uptake these lipids. And what we did is to study these mutants in this context. That's how we got into this knowledge by using these 3 mutants that I presented earlier that have key functions in these circles that you see there, which are the key steps in the modification of the lipids. And the mutant phenotypes, as I mentioned, differ to the wild type that is highly branched. And in order to do this study, what we did was to generate, study the lipids by using lipid profiles. What we did was to harvest tissue that was colonized by the fungus, which will have both plant and fungal tissues, so that we can study both the plant and the fungus at the same time and figure out what's going on with the lipids in this case. And what I'm going to do in this series of slides is just going to summarize the knowledge that we got from that and why these lipids are important. So first of all, in a normal situation, what happens is that you have this flux of lipids that follow these blue arrows going to produce these types of lipids, the membrane lipids and glycerol lipids. However, in the context of this symbiosis, there's going to be a boost in the production of these lipids that will then divert it to produce the monoacyl glycerols and to be uptaken by the fungus. So what's going to happen is there's going to be a transfer of these lipids from the plant into the fungus. And what happens then if we have one of the mutants, for example, the FAT-M mutant? If we don't have that mutant, then what happens is that there's a reduction in the production of these free fatty acids, and we know that, and we can measure that, and specifically we see the 16 carbon molecules, the lipids that are reduced, and also the concomitant reduction in the lipids that go into the fungus, which we can also measure. So the fungus doesn't get the lipids that it requires to grow. And there's the collapsed arbuscule phenotype that I showed earlier. Next, what happens if we don't have the RAM2 mutant that is shown there is that this enzyme is the one in charge of producing the MAG lipids, the monoacyl glycerol lipids. And what we can see is that there's a reduction in the production of these lipids as measured in the graph on the right. Interestingly, though, instead of seeing an accumulation of the acyl lipids, what we see is a diversion in the flux of the lipids that go to create most phospholipids, which specifically accumulate in PC and PA lipids in the plant. So it's kind of an indirect way of measuring what's going on with these types of lipids. And finally, we have what is the main transporter that moves these lipids to the fungus, and the candidate is the STAR gene, an ABC transporter. And what happens is when you don't have this ABC transporter, what you will see is an accumulation, or what you would expect is an accumulation of these monoacyl glycerols in the plant. However, because of the way there's this feedback between the plant and the fungus, it's more difficult, because when there's no feed of these lipids to the fungus, then the symbiosis doesn't take place, and therefore there's a whole reduction of the induction of all these other lipids, and the whole thing goes down. However, what we can do is compare the different mutants, and if we compare the STR mutant, the STAR mutant, versus the other RAM2 mutant, we can see an increase in the accumulation of these lipids, which tells us that indeed those monoacyl glycerolipids, specifically the 16 carbon molecules, are being accumulated in the mutant. And which is an indirect, I mean, still is an indirect piece of evidence, but suggests that maybe that the STAR ABC transporter is involved in the transfer of these lipids. And further studies by other groups later on also provided more direct evidence that there was indeed this transfer of lipids. So even though we were the first group to provide the evidence for this transfer, the direct evidence actually came from other groups. And even also recently, it was shown that not only in flowering plants, but even in bryophytes, in the liverwort Mercantia, this transfer of lipids is still conserved. So it's very important for mycorrhizal symbiosis. And that has basically changed the paradigm, an old paradigm that we had, in which we thought that the main form of carbon transfer from plants to fungi was carbohydrates. And now we can change that. Lipids are very important in this context. So just to summarize, what I showed you is all of it, basically, is that phylogenomics is a very powerful tool for the discovery of new genes, and that some of these genes work as part of a module that regulates this lipid pathway, synthesis pathways, and that, of course, during mycorrhizal symbiosis, the exchange of nutrients is important, and specifically the exchange of lipids from plants to fungi is very relevant. And finally, I just want to kind of end up with a slide in which I was showing the genes that have been identified, in which this is the state in which we started the study many years ago. Eventually, it moved into something like this, so kind of many more genes being identified. And the whole purpose of the study that of the research that we do is to eventually get to a point that looks something like that, like completely filled of all these holes in knowledge that we have. And most importantly, of course, know not just what are the genes, but what are the function and how do they relate to each other. So now there are multiple questions now that are still being unanswered. And as Omar mentioned, now I just recently moved this year to the Danforth Center, which I started my group, and I'm going to continue working on mycorrhizal symbiosis, trying to figure out what are the functions of these multiple genes and what are the mechanisms for the transfer of these nutrients. So finally, I would just like to acknowledge the people that participated in the research that I presented today, and also the funding sources. And finally, also I would like to acknowledge my roots, as I was saying earlier, my mentors, which helped me so much in my career and have been so important in becoming a scientist. So I really appreciate my time at IBT and all the knowledge that I got from that. And with that, I would be happy to take any questions if there are any.
Are there questions for Armando? Vasily?
Hi, Armando. Nice talk. I just want to, partially my question was answered in your last slide, but my question was if you find many genes that goes to plastids. And I saw that you have several, and I saw that you have several CCDs. Which is quite interesting, because I was wondering about the mycorrhizin in finally is an carotenoid, so a way to affect this is through affecting the exchange in signals. So could you comment a little bit more if you have many other putative chloroplasts and where they go?
Yes. So just talking about the genes that are conserved for symbiosis that I talked about, because there can also be many other genes that are not in that group. There's a few that have been identified as being just in plastids. One of them is a novel CCD gene that was identified recently in, I forget if it's rice on a different plant, but they actually characterize the compound that it creates, and I think it's called cyosine. It's a different compound that also is involved in symbiosis and also in, I think, rhizobial symbiosis. And there's also other enzymes that we know are related to lipids, because plastids are so important for lipid production that we know that there are multiple enzymes that are involved in either modification of these lipids and maybe other things, and also carotenoids, as you mentioned. So definitely multiple genes, but we're still kind of learning which ones are those, and yet there's not so much known other than this CCD gene that I told you, and a couple of P450 enzymes that have been identified.
Hi, Omar. Armando, thank you for your talk. It was very nice. I really enjoyed your talk. I have two questions. The first one is the PT4 that is highly induced during the mycorrhizal association. Does it always correlate with the high affinity ones for the phosphate trap?
Does it correlate to what you said, sorry?
To the high affinity transporter.
Yeah, actually, I think this is not such a high affinity transporter. It's in the family of PHO1, like of the transporters, but because the concentration of the phosphorus is so high in our buscules, like you can imagine like a really concentrated, a cell concentrated with a lot of phosphate, it doesn't require that much.
Okay. So you need more like the low affinity transporter there?
I think, well, so in this case, so in those cells that are colonized, there's all these other transporters that are not just the PT4, but the main one is this PT4, the one that is actually taking up the phosphorus from the fungus into the plant. So it's that specific one that works. So all the others, even though they are expressed, they don't seem to have an important role for the transfer.
Okay, thank you. My second question is, it's well known, for instance, that the fungi requires a lot of lipids, including the very long fatty acid. And that's why they also require a lot of these components from lipids coming from the plant. My question is, do they need all these lipids for the arbuscle formation or for the actual functioning?
So that's a very, very good question. And it's kind of difficult to separate the things. So to separate the formation of the membranes, there's lots of membranes being formed in the arbuscles, as you saw, like all this branching of the hyphae. Of course, there's lots of lipids that the fungus requires, and also the hyphae is full of lipids that they store there. Mycorrhizal fungi are very well known to be full of these triacylglycerol lipids, like these reserved lipids, and yet they lack the main enzyme, the fatty acid synthase, that produces de novo fatty acids. And that's one of the first clue that we had of why they require the lipids from the plants. But whether they need it for membrane formation or the functioning of the fungus for energy, or even for signals also, I don't think we can separate them. I think the most likely answer is that they need it for all of those things altogether.
Thank you.
¿Puedo preguntar?
Sí.
Hi, Armando. I'm Elena.
Hi,
thank you very much for your kindly words. We have a really good time during that time. I was wondering if you have any idea about which are the molecules that the fungi are in which the fungi are regulating the lipid biosynthesis. It's not the phosphate, it's, well, you say something about signalings, but do you have any idea what is going on?
So if you are talking about the signaling that takes place once the arbuscule is being formed, we don't know much about that kind of signaling yet. and particularly in the case of the transfer of phosphorus, we don't know how is the plant cell like detecting all of these different signals. There's multiple signaling cascades that we know are activated inside the arbuscule, and recently there's been a few kinases that have been identified, like signaling kinases, and there's been ideas of being peptides that are being signaled from the fungus to the plant. Maybe it's also carbohydrates, like cell wall components from the fungus that the plant cell can differentiate between pathogenic fungi and symbiotic fungi, but yet we don't really know. It's been more studied the initial signaling that takes place before the contact between the fungus and the plant, and there's some like lipoketo-oligosaccharides that are known to be signals for the plant, but once they are inside in the cell, is not so much known.
Okay, thank you. Great work.
Thank you.
Well, a quick question, Armando. What type of ABC transporter is the one? Do you know that?
What kind of ABC transporter?
You identify the type of ABC transporter that is involved in the transport of lipids?
Yeah, so the one I was talking about, the STR or STAR as we call it, it's a half ABC, so family G transporter. It actually generates like a full-length ABC transporter with another gene called STAR2. And this is from as I said, so family G of ABC transporters. And that's only, so far there's only indirect evidence, even the one that I presented and other studies that have shown that kind of put that transporter as the one responsible for transferring the lipids, but we don't have the direct proof yet. We think that most likely this is the one. And there are other ABC transporters from different families, particularly family B of ABC transporters, that are also involved in symbiosis, but they seem to be involved in different things, like maybe even auxin transport. That's a recent study was showing something like that, and maybe other things, but we don't know exactly. So far, the best candidate for the lipid transport is this STR star transporter from family G.
Okay, thank you very much.
Thank you.
And thank you, Armando. I think we will move on to our last talk. So
thank you.
We can skip the feed of the Zoom. And I'll let Patty Leon introduce our next and last speaker.
Hello, everyone. And it is really my pleasure to introduce our last speaker, which is Jeannie Walbot. And probably for many of us, Jeannie doesn't need to be introduced really, because we know her very well. But for those people that do not know her, I want to mention that she is a full professor in the Department of Biology at Stanford University, and also honorary adjunct staff scientist at Carnegie Department of Plant Biology. Jeannie has been and is one of the persons where plants is really a passion. She is a passionate of all type of plants, but in particular, I think her heart has always been maize. And Jeannie has done different research in maize that goes from transposable elements. And more recently, her interests have been focused in understand the differentiation of maize anthers. And he has been working with the small RNAs and trying to understand his function, the particular this fatty RNAs in anther development. And also, she has been working recently in trying to identify different transcription factors involved in anther development. So I think we will hear about this, but before ending, I also want to emphasize the fact that even though that Jeannie is a pretty busy person, he has always been very, very supportive to Mexican scientists. And I think she has collaborated with many of us, particularly in the IBT, and she has also participated in many aspects of the development of this institute. So I think it's an honor that she accepted this invitation in the 40 celebration of this institute. And without any more words, I leave you with Jeannie. Thank you, Jeannie, for being here.
Great. Well, of course, it's a great honor for me to be invited to speak to you today. And I wanted to start by acknowledging all of the fantastic memories I have of many, many trips to the IBT over the years. I'm pretty sure I was there the very first year and the second year, and then maybe every 3 or 4 years since then. So I've watched your institute grow and thrive. And I'm very proud, as I know all of you are, of the progress that you've made over the years. Today, as Patricia mentioned, I'm going to be talking to you about our recent work on anther development in maize. And this is a direct outgrowth of our 25 years working with mutator transposons, as we figured out 15 years ago that the transposon was activated in a very particular way during anther development, which led to the high frequency of mutations through the male germinal cells as opposed through the female. So get my slides organized here. So today, what I'd like to do is to concentrate on work that we've performed using single-cell RNA-seq of maize anther cells. I'm going to introduce a new technique that we've perfected, and actually announced this technique, the beginnings of it, 4 or 5 years ago at a meeting a few years ago in Merida, and then talk about germinal cell development, and then our most recent work on the developmental trajectory of all of the anther cell types. So in order to be prepared to learn more about anthers, you need to know a little bit about them. So individual maize spikelets have a pair of florets. The upper floret anthers are about a day older than the lower floret anthers. And in our work, we generally use only the upper floret anther. And when we dissect these, we put one in the freezer to keep for future studies. One is prepared for microscopy, and one is used for immediate molecular biology, whether it's proteomics or transcriptome analysis. And then if we look at a transverse section of an anther that's about to enter meiosis, we can see all of the cell types. So there's an epidermis surrounding each of the 4 lobes of the anther. The subepidermal endothelial cells have chloroplasts in them, and after meiosis, they become photosynthetic and help support the development of the anther. The red zone here, the middle layer, is kind of a mystery zone. Some species of plants, flowering plants or gymnosperms, have a middle layer. Some have 2 middle layers, and some have no middle layer. So it seems to be a dispensable layer. Maize happens to have a single middle layer. And those cells are sister to the tapetum, which is a really important cell type in the anther, because the tapetum makes the nutrients that support the germinal cells, which start their life as archosporial cells. So those are mitotically active cells. They become myocytes, so capable of undergoing meiosis. They give rise, each myocyte gives rise to 4 haploid gametophytes, and those develop individually into the pollen grains. And their nutrition, as well as most of the components of the pollen wall, are synthesized by the tapetum and secreted into the space occupied by the central germinal cells. So in addition to the 4 lobes, there's also a region called the connective, which is filled with relatively undifferentiated parenchyma-like cells, and including xylem and phloem, filament, vascular system that supplies the nutrients to the developing anther from the body of the main plant. Oops, sorry. So we had done a lot of work with standard transcriptomics, but why did we need to switch to single cell work, which has been challenging? So our initial motivation was because we really wanted to understand what was going on in the archosporial cells as they developed by mitosis and then switched to becoming myocytes, because it's during these stages that our mutator transposons were activated. The archosporial cells are actually a rare cell type in the developing anther. They're only about 1% of the cells. And so it was going to be difficult ever to tease out of total transcriptomes what was going on in this cell type. Also, people who study meiosis had developed various methods for recovering myocytes. So we felt it was possible to acquire them and therefore to be able to do transcriptomics of this cell type and to push it further to get individual cells. We've now moved on and reworked our methods so we can study all of the anther cell types, even though they are much smaller and contain far less messenger RNA than the giant germinal cells. So really important features of single-cell RNA-seq that distinguish it from standard transcriptome profiling is, as a first step, all of the transcripts in a particular cell are given a short barcode. We use 6 bases. So that identifies later in the transcriptome data the cell of origin for each transcript type. And this also allows the pooling of thousands of cells in one lane of a sequencing run. Then after all the transcripts in a cell get a barcode, then we also barcode each transcript in the cell. So this is a set of random barcodes attached to the cell identifier barcode. And this transcript barcode is usually called a unique molecular identifier, or a UMI. And then later, when you're reporting transcript counts, abundance, or the presence absence of a transcript, you count each UMI only once. So you're basically saying, at the time I isolated the cell, this molecule existed, this particular mRNA existed, here's its unique identifier. And because there's a lot of amplification steps in single-cell RNA-seq library preparation, you only want to count that transcript one time, even if you recover it multiple times in the sequencing, in order to be quantitative about the mRNA content of that original cell. So in our work with archosporial cells, as I mentioned, we start with the upper floret anthers, and we were interested in the range of developmental stages from when the archosporial cells are first specified. So that's in about an 0.15 millimeter anther, 150 micron anther, through the leptotene-zygotene transition during meiosis. So in transverse section, this is what the very immature anther would look like, with in this kind of view, 1 or 2 archosporial cells. By the time you're in meiosis, in transverse view, you would have 5 myocytes. So after we dissect the anther, we stage it. This is based on length. We make protoplasts, or partially protoplast the intact anther. And then we can identify the archosporial or pollen mother or myocyte cells because they're so much larger than the somatic cells. So in a microscope view, you'd see these rare, very large cells and lots of the small somatic cells. And for this initial study of the germinal cells, the postdoc Brad Nelms picked these large cells up manually using a needle. And so he took a picture of each one, a baby picture, and he could incubate them briefly before distributing them into a plate for lysis and library preparation. Now, because these cells were so big and we knew they had so much RNA, we tried kind of a risky, but it worked out well, procedure where each cell lysis was split into 2 technical reps. And we did this because no one else had utilized a split cell protocol to try to find out how good the single cell library preparation method really was. So it's quite lengthy, and there's really a lot of steps where you could imagine biases occurring in terms of what messenger RNAs are copied by reverse transcriptase, how they're incorporated into the final sequencing cocktail, et cetera. So it had really been a very major worry in the field. And we thought because we had such RNA-rich cells, we could try to address it. And that's what's shown here in panel C. I also want to point out that this entire study was done with 144 cells. So some people are under the impression that to do fruitful single-cell RNA work, you need tens of thousands of cells. That's not true. This is not a really large number of cells, but we answered in a series of important biological questions. So in this diagram, you're looking at a principal components analysis, and the gray scale of the dots tells you the anther length, so the approximate stage of the anthers. And you can see that the cells go from this light gray through a steady progression. These are archosporial cells, then pollen mother cells. So those are post-mitotic archosporial cells. And then this jump is to the myocytes. So that's already telling you that there's going to be some difference in the transcriptome between myocytes and their precursors. And then these dumbbell shapes are the 2 cell replicates. So you can see that cell replicates are very close to each other, not necessarily identical, but really close. And in fact, when you plot replicate 1 against replicate 2, the R squared is 0.97. So despite all the worries in the field about the procedure, if done appropriately, if done correctly, replicates are very, very close, if not nearly identical. Now, a convention in the field, instead of using or referring to principal components plots, even though that's the underlying methodology, is to refer to this trajectory of how cells are developing as pseudotime. So from the beginning to the end of your study, you connect the dots basically, and this gives you a sense of how the cells are changing over time. And a kind of technical or analytical innovation that we made is we noticed when you plot the cells ordered by
del grupo de los hilos de entrada a los sistemas operativos. Entonces te voy a enseñar a cómo repararlo. Para ello vas a necesitar lo que Windows 7 en la descripción del video. Y bueno, vamos a esperar que esto se trabaje. No importa, pero bueno, por costumbre vamos a ponerle español México, reparar el equipo. Y bueno, aquí nos va a empezar a... Dice buscando instalaciones de Windows, en este caso nos tiene que encontrar esto, de hecho ya lo encontró. Vamos a darle en siguiente. Bueno, en un momento ahorita que acabe. Si presentas problemas en lo que es el Windows, te va a pedir que lo repares. Ok, bueno, aquí vamos a darle en siguiente. Nos vamos a, podemos ocupar
shows us right here at the archosporial cell to post-mitotic pollen mother cell stage, there's definitely a blip. So there's going to be new transcripts and or the loss, massive loss of transcripts that pre-existed. But even more dramatic were two major changes in the transcriptome that are attributable actually to new synthesis, as I'll show you later. One occurs approximately at mid-leptotene, and the second one at the leptotene-zygotene transition. And this is of interest to people who study meiosis in plants because the leptotene-zygotene transition has often been identified as a checkpoint, a place where meiosis gets a go or no-go signal. And so it's likely that the biosynthesis of all of these new transcripts is important in that decision, in that checkpoint of whether the meiotic cell is sufficiently normal to proceed through the rest of meiosis. So that was an exciting output from this study. I'll just reiterate things that to someone like me, a developmental biologist, were particularly interesting, that during the mitotic proliferation phase of the archosporial cells, there's almost no change in the transcriptome. So these cells are basically identical during this entire transient amplifying stage of development. And we also demonstrated in data I'm not showing you that all of the archosporial cells that were born back here at the beginning all undergo 3 to 4 rounds of mitosis. So this settled kind of a long-standing problem of whether there were some archosporial stem cells that proliferated a lot to generate the population. We're going from about 10 or 12 starting cells to about 150 to 160 at the end. Could those have been generated by a few stem cells, or did all 10 to 12 of the starting cells undergo mitosis? And that's what we found. And their transcriptome stays pretty much constant. When they cease mitosis, they synthesize a number of new transcripts. And then the exciting finding at leptotene and the leptotene-zygotene transition of many, many new transcripts. Oops, sorry. Okay, so the study I just summarized for you was published in Science in 2019, and it naturally led to study number 2, which was what happens during the rest of meiosis and beyond, all the way out to pollen grains. And the primary question in the field was, after meiosis finishes, when does the haploid genome express new transcripts? And the assumption, and it was a good one based on yeast and primitive plants, was that the haploid genome would be expressed immediately after meiosis, because of course, you're now a haploid gametophyte, and presumably a gametophyte needs to express different genes than a diploid sporophyte. So the assumption in the field that was for angiosperms that haploid gene expression would start at the conclusion of meiosis. What we actually found is the transcripts that were made at the leptotene-zygotene transition at the beginning of meiosis persist through the uninucleate microspore stage. So the whole time the gametophyte has just one nucleus. And that means that these transcripts made early, leptotene-zygotene, early in meiosis, last through the rest of meiosis, last through the tetrad stage, and for the 10 days of the uninucleate microspore stage. That's all this red. There's definitely transcripts that are disappearing. What's important is there's almost no transcripts that appear during the haploid phase of this first 10 to 11 days of what is close to 20 days till you get to pollen. So more than half of the haploid development is supported by transcripts that were made weeks earlier. And the way that we could figure this out is we crossed 2 well-studied inbred lines of maize, A188 and B73, to get F1 hybrids. And the reason making this hybrid was important is each of these inbreds has a really very good genome, and the two inbreds differ by more than 1% in terms of their bases. That is, there's quite a bit of SNP, quite a bit of polymorphism in the genes. And so for most alleles that we would sequence, we could attribute that allele to either the A188 or the B73 parent. We were sequencing about 200 bases, and we expected 1% SNPs. So we typically had 1 or 2 or 3, 4 SNPs to do the assignment to A188 or B73. So when we used data from cells isolated from before meiosis, we saw that there was basically even representation in a single cell of the alleles from both B73 and A188. There are a few alleles, a few genes where all of the sequences were from one parent or another. And the vast majority of these turned out to be cases where the gene was actually only present in one of the two inbreds. So these two inbreds of maize differ about 5% in actual gene content. And so those inbred specific genes contributed to the 0 and 100% cases. When we looked at haploid pollen, which is the end product of the developmental trajectory, we saw that individual pollen grains, allele by allele, expressed either the A188 or the B73 allele. And that's exactly what you'd expect from a haploid. Of course, this haploid pollen is the result of meiosis, which means recombination. And so we have both A188 and B73 alleles present, but at any one locus, there's only representation of one of the two parents. And so if we map these alleles onto the chromosomes from just one pollen grain, we can see here for 3 chromosomes that on chromosome 1, the start of the chromosome, the tip of the chromosome, is all A188 alleles. And then there's a sudden jump, as you would expect from recombination, up to the B73, which continues across the centromere. And then partway through the long arm, boom, recombination. Now we have the A188 allele, and we had a second recombination on this long arm back to B73. So for each pollen grain, we could map the inferred positions of recombination. They were different on every chromosome for every pollen grain, just as you would expect. And then as a final sort of proof that we hadn't lost anything during meiosis and recombination, we pooled a group of pollen grains and sequenced them. And we basically recapitulated the pattern that we see in diploid precursors. So everything is still represented, but when you go to single cells, you see that representation is basically one parent or the other. And of course, the Big Bang finding was that these developing pollen grains depend on sporophytic transcripts for more than half of their development. So looking at this, again, I really want to reiterate this, that in meiosis, the tetrad stage, and the uninucleate microspore, we have biparental, biallelic expression. And so when we plot allele representation, we see representation from both parents. But as soon as you get to the binucleate stage, so now we have a vegetative nucleus, vegetative cell, and a generative cell. This generative cell will later undergo mitosis to make the 2 sperm. As soon as you get to this stage, you have the monoallelic expression. So it seems to be associated very closely in time with the mitosis that generates the two cell types of the binucleate pollen grain. We're not sure if the transcription occurs before, during, or immediately after the mitosis. That would be of interest because many people assert that the vegetative cell is transcriptionally active and the generative cell might be transcriptionally silent. So we would like to use this methodology to figure out if the transcripts that we observe that are clearly from the haploid genome are in fact all being made by the vegetative nucleus or whether the generative nucleus is also participating. Now, another thing that we could do with these data that addressed an assumption, an important assumption about plants, for which there is some data, that is that plants exhibit a lot lower genetic load than animals. So in animals, the gametes, the sperm and the egg, are pretty much transcriptionally silent. There is some new RNA synthesis, but it's very modest. And as we know, inbreeding in animals leads to the exposure of many recessive deleterious alleles. So it appears that there's not really a good system in animals to eliminate deleterious alleles in the haploid generation. The opposite is true in plants because the gametophytes, the haploid phase of the life cycle, is genetically active, making transcripts, expressing genes. And in lower plants, below ferns, the haploid phase is the dominant phase of the life cycle and photosynthetic. So much of the genome is tested for quality in the haploid phase. So we were interested in quantifying the expression of genes that are found expressed somewhere in the sporophyte and in the gametophyte. And that's this really big dark blue area of this pie chart. And then of the genes that we identified expressed at any point in the germinal cell lineage, there were only fewer than 2,000 that were gametophyte specific and close to 3,000 that were sporophyte specific, that is expressed in the pollen mother cells, but the transcripts were lost, not present, and not re-expressed by the time you got to haploid gene expression. Another finding from looking at these studies is we used standard methodology from evolutionists to ask whether genes expressed in the sporophyte versus the gametophyte were under purifying selection. And very rewarding to us, the genes that are expressed after the pollen mitosis 1 and or in mature pollen are definitely under purifying selection. There's no significant difference between the once haploid expression is activated between that initial haploid expression and pollen-specific expression. All of those genes are under purifying selection compared to genes that are only expressed in the sporophyte. And so we can conclude that many haploid expressed genes are also expressed in the diploid sporophyte, and thus haploid selection can act if the gene expression in the haploid is important for haploid life. There will be a quality check and therefore an explanation for the reduction of the genetic load in the vegetative plant. How much more rare deleterious mutations are, say, when you self a plant. You generally don't see a lot of really sick plants. The exception in angiosperms would be that you get lots of albinos, because in the haploid gametophyte of angiosperms, there's no test of photosynthesis. And so that is a form of genetic load and explains the beautiful variegated ivy and other kinds of variegated plants that we see all around us. Okay, so these studies of the germinal lineage were super successful. And yet I need to tell you that they were really, really tedious to do because each cell was picked up by hand. And in the original method, because of the protoplasting that was involved, we made a time limit that we could only harvest cells for 90 to 100 minutes after we took the tassel off the plant. And that meant that we could recover, and by we, I mean 9 cells recovered by Brad and 1 cell by me. It's really difficult to pick them up. So in a tedious day, we could recover 10 cells. And it actually took almost 6 months for us to get all 144 cells that were in the first study. While we were doing that study, we were trying other methods. A Stanford lab had a biosorter, which was a machine invented to sort egg cells from nematodes, from Caenorhabditis. And the machine is pretty expensive, and we were being charged $50 an hour to use it. It's really, really hard to clean. And we had to work in another building, which meant much longer time between getting the cells and collecting them. And basically, during the pandemic, we weren't even allowed to go in the building. So that was a no-go. We also tried fluorescence activated cell sorting because we can stain our nuclei with cyber green, so we could make them green. And at Stanford, we have to pay $70 an hour to use the machine, and we're not allowed to use it ourselves. So we have to pay someone $60 an hour to do our work for us. And even at the lowest flow rate, we were shredding up the big cells. So this was really useless for our cells. And finally, we contemplated, but never actually did 10x genomics, a microfluidic device, which is probably the most common method used in plant single-cell RNA-seq. Mostly people use nuclei from protoplasts. It's quite expensive. It's a monopoly kind of situation in terms of the device and the kits. And for us, it just lengthened the time between harvest and getting the cells. So we were frustrated, I think, that we really needed a much better method, like just rethink what we're doing. And we came up with a novel approach that tried to address the 2 big challenges in plant single-cell RNA-seq. The first is the protoplasting part. So you need the cells in standard protoplasting to remain viable and fresh during the digestion. Of course, the cells change during digestion. You're ripping off the cell wall. After you've protoplasted, the cells lose all of their morphology, so you can't really identify cell types. And the worst is that for every tissue, organ, and species, you typically need quite a bit of optimization to get good protoplasts that have intact mRNA for single-cell RNA-seq. So what we have done is invent a new protocol called FX-Cell, and it's published in bioRxiv, and it's in review right now. The main innovation is immediately on dissecting the anther, we fix the tissue. So no more changes to the mRNA, no more changes to the cell. And so there's just a few minutes between dissection and stabilization of the RNA. So that we thought was a big win. When we actually recovered cells from anthers and a lot of other kinds of tissues, we found that there was a big enhancement in cell recovery. So a typical protoplasting method releases only 5 to 10% of the cells. You get a big enrichment for some cell types and absolutely no examples of other cell types. This FX-Cell protocol gives you a representative tissue sample. It's been successful with almost everything we've tried, except cells that have really, really large vacuoles, because those tend to pop. and disrupt the cell during the fixation. And another innovation we've added is to improve RNA integrity. We've removed most of the fungal ribonucleases that are in the protoplastin cocktail that everyone uses. And that's actually pretty straightforward with GMP agarose beads. These fungal ribonucleases are of an entirely different family than the typical ribonucleases that all of us are familiar with from bacteria or mammalian cells. And they're not inhibited by any of the standard anti-RNase agents. So getting rid of them is the best method. So in this protocol, our old protocol was take an anther and incubate it with digestive enzymes, usually for 60 minutes or so. And then the way that we disrupt anthers, and this also works for leaves and lots of other organs, is a method that Paul Green invented a long time ago. You just put your tissue between two glass slides and put a piece of tape on the end of the slide so the slides don't quite touch each other, and then just rub the two slides together 3 or 4 times, and the cells are liberated. In our new protocol, boom, we go right into farmer's fixative, which is 3 parts ethanol, 1 part acetic acid. You can fix for a long time or a short time, under an hour. We wash them and then put them in phosphate buffered saline and add the enzymes, which have been purified now to get rid of the ribonucleases. We use about 1/10 as much enzyme. And the big innovation is because the cells are now fixed, we can incubate at the optimum temperature for these fungal digestive enzymes. So the fungi that yield the cellulases and macerozyme are the fungi that decay wood in the forest. And if you've ever felt the heat rising from decaying wood, or if you have a compost bin at home, you know that the temperature gets up to about 50 degrees C, and that's actually the optimum temperature for those digesting enzymes. So we can use much less enzyme, a much shorter time than incubating fresh tissue at 30 degrees. So we kept under our 100 minutes to recovery of the cells here on the fresh side, but on the fixed side, we don't need to be in such a rush because nothing's going to change after fixation. So now looking at the whole anther, we are getting more ambitious. We want all of the cell types. Here's a little schematic of the protocol. Dissociate the cells, and then we stain with cyber green so we have fluorescent nuclei, and we filter them through a 40 micron strainer to get rid of this kind of junky cell wall stuff and any cell clumps that didn't turn into protoplasts. And then I'll show you later a new machine that we use to separate the cells into plates. So before we started, we had data from an earlier study where we knew at each stage of anther development what the composition was in terms of cell types. So the archosporial cells, here I'm showing you at approximately mid-meiosis, they're about 1% of the total. The connective and vascular cells are a little over 12%, and then the other cell types by color code. By far the most prominent cell type are the tapetal cells. So they're almost a third of all the cells. And down here in panel D are the shapes and sizes of the 5 lobe cell types. So we have 2 columnar cell types, epidermal cell type and endothelial, small rectangle, middle layer cells, cuboidal, tapetal cells, and then the really giant, drawn them here as a cube, but they're kind of rounded myocytes. And so after we digest, we can look through a microscope and actually identify cell types based on shape and to some extent the properties of the nucleus. So Here's an epidermal cell, wider and longer than the endothelial cells, which are very thin, middle layer cells, about half the length of an endothelial cell, cuboidal to pedal cell. And by this stage of development, they are binucleate. So that's a real indicator that you've got a tapetal cell, very unusual cell phenotype. And here's one of the large pollen mother cells, which is not shown to scale. It would be considerably larger than this picture. And we also found the vascular and connective cells in approximately the correct proportion. So in our early studies, all the studies with the germinal cells, we were releasing about 5 to 7% of all of the cells in the anther. With the fixation protocol, we get 82 to 96% release. So this is awesome if you're trying to study all the cell types, because we finally have all the cell types. We also compared this fixed cell protocol to the optimized protocol by the sort of best people in the field, Ken Birnbaum at NYU and David Jackson at Cold Spring Harbor. So they worked very hard to optimize a protocol for maize root tips. And here's their result. They were releasing just over 20% of the cells, or sorry, the number of cells released per root tip, about 20,000. We're getting over 100,000. So about a 5-fold improvement. And by microscopy, we're getting all the cell types, and they were basically only getting the epidermis and subepidermal cells. So the new method, harvest the tassel, dissect and fix the anthers, less than 10 minutes to get stable RNA, get single cells, stain with cyber green. We found it easier to collect the cyber green stained cells, so we could get up to 20 to 50 cells collected manually in half a day, but still kind of sad. So a startup company in the Stanford Industrial Park called HANA invented a new machine called NanoCell. And this can collect 96 fixed cells in 2 minutes. I mean, that's a world of difference. And 384 cells in less than 10 minutes. 95% of the wells contain an actual cell and not some kind of debris. So the secret to this NanoCell is it's almost a gravity feed machine. There's only 2 psi of pressure, and we could never achieve that in a fluorescence activated cell sorter. This uses the same 2 common lasers that are found in an expensive fluorescence activated cell sorter. So you can sort based on fluorescence, or you can sort based on cell size. It's by far the fastest cell isolation machine on the market. So it's actually faster than a fax. It just has a capacity for only a few thousand cells, and then you have to reload it. So it has 2 sorting modes, both of which we've used. You can use single cell sorting, or you can use an enrichment mode where you give it certain parameters, for example, the description of the size of an archosporial cell, and that allows you to recover a rare cell type. High recovery rates, so it's ideal for really small starting samples, as few as 100 cells or less. The other feature that's kind of handy is you can program each well to get a specific number of cells. So on a plate, you might want to collect in 90 of the wells individual cells, and then the last 6 wells maybe, quote, a representative sample of 50 cells each for standard transcriptome profiling that would be emulating what a whole anther was like. So the machine uses custom plastic cartridge. They cost $85 each. And the company, of course, tells you that you can only use them once, but we've used them up to 10 or 15 times with no problem. And you load your sample up here, and it flows downhill, basically by gravity, and it's imaged in a little area down here. The machine is about the size of a toaster oven, so it's really small. It has no instruction manual. You flip up the lid and there's an LED screen that tells you what to do. So it takes about 5 minutes to learn how to use it. You wash the channels through the machine with 2 mLs of fluid, and then you load your cells and start collecting. And then make your RNA-seq library. So we're really proud of this. The cell fixation decreased our time to stable mRNA by 10-fold. The NanoCell machine increased our efficiency in collecting cells by 100-fold. So from an engineering perspective, we made a 1,000-fold improvement. And this has really facilitated all the studies that we do now. And particularly, it's eliminated personal fatigue from trying to collect individual cells under a microscope. And it totally eliminated the person-to-person variation in cell collection success. So it's a smooth path from tissue to cell recovery. So the current big project in the lab is postdoc Billy Marchand. So he's working on developmental trajectories for each of the low cell types, as well as we're trying to figure out the connective and vascular cells. And a couple of our critical questions are, we want to understand the separation of sister cell types. So the middle layer and the tapetum come from a periclinal division of a stem cell, a secondary parietal cell. So they're born as sisters, and we want to figure out how they develop differentially after that. And the method here has been to collect from 11 stages of anther development, 4 plates of 96 wells each. So start with 384 cells from each stage of development. And just to remind you, we're interested in each of these cell types that are present in the anther from. So in our very first run, 384 cells, we had 350 cells with usable data. And just plotting the most highly differentially expressed genes, we could cluster the results into 5 apparent cell types. Now we have more than that cell types to start with, but we knew we probably wouldn't get enough data for myocytes for this kind of analysis because they're only 1%. And we did, I'll show you later, find 3 or 4 myocytes in every sample, but in this heat map type display, they're not going to show up. So we have the tapetal cells, which are the majority, epidermal cells, endothelial cells, we hope middle layer cells, and then the connective and vascular cells seem to go together. So a convention in the field is to use what's called U-mapping, or it's a long word. It's just a statistical program for clustering. And we've used multiple methods to identify cell types in the clusters that have names. We knew a lot about the myocytes. They're rare cell types, but super easy to identify. And because they have so much RNA, we get a really strong signal from them. We knew that about the endothelial cells because they have developing chloroplasts from a really early stage in development. So enough light penetrates into the developing tassel, into the developing anthers to stimulate chlorophyll production in the endothelial cells. So we have lots of marker genes for that cell type. And through genetics, we had a number of marker genes for the tapetum, lots of transcription factors, and these tapetal markers were validated earlier by in situ hybridization. And we have markers for the epidermis because it makes wax in a cuticle and has other, we use some transcription factors that were epidermal specific. And that leaves us with these 2 kind of perhaps 2, perhaps 4 here, 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 and here clusters. We're hoping over time to be able to assign phloem, xylem, connective, which are parenchyma-like cells, and the middle layer to these units. And as I mentioned, we use marker genes. So we know marker genes, say a tapetal marker gene. We have recently published a laser capture microdissection study where we captured separated tapetal cells and myocytes. So we took markers that were highly enriched in the tapetum and mapped those onto the cell clusters. Here's the tapetum. And in this smaller diagram, this is the zone that really lights up with those tapetal specific markers, and ditto with the myocyte markers down here. And we do that for other markers. Sometimes we get surprises like MS8, male sterile number 8, a gene that we cloned. We knew it would be expressed somewhere in the anther, and the in situ hybridization was a little bit ambiguous, but seemed to be middle layer. But when we mapped expression, it was kind of minimal almost everywhere except in some tapetal cells. And here's another disappointing marker, MS44, which we thought was tapetal specific, but clearly shows up in lots of cell types. So we've tested about 50 to 100 genes, and a subset seem to agree with all of the other biology about where they're supposed to be expressed, and the other half turn out, at least in this method, to not be very good cell markers. So another way of considering these data is, I've usually been showing you sort of mid-meiosis diagram. We can also plot all of the cells, now color coding by the stage of development. So this sort of reddish and tannish marker are from very immature anthers, including also the 1 millimeter stage. That's when the pollen mother cells are present, but the other cell types are still mitotic. And you'll see that most of those colors are here in this big central blob. So we contemplated this for a long time, and our current working idea is that although by microscopy it's so easy to identify these cell types, through even the 1 millimeter stage, these cell types are not yet expressing differentially expressed marker genes that say, I'm a tapetal cell or I'm an endothelial cell. But as soon as we get into this green zone, we can see here's the beginning of the tapetum, the green at 1.25 millimeters. And by here, this more aqua area, slightly aqua over here, is after meiosis. And here's at the binucleate pollen stage, the tapetum is really distinctive. So tapetal cells exist back here at 0.7 and 1 millimeter, but they're not distinguishable as a unit. But boom, all of a sudden, once they develop specific tapetal differentiated functions, we can clearly see the trajectory of the tapetal cells. So the only two cell types that are, oops, I didn't mean to do that. The only two cell types that are distinguishable kind of at all stages are the epidermis here and the germinal cells. So they're epidermal cells, you can see brown and orangey things in here, as well as things from all stages. So the epidermis, which of course is from a different layer of the meristem, and I guess we would consider separate from the beginning, and the germinal cells are born about 2 days before this stage, and they already have a separate identity compared to the somatic cells. So I think a big take home for us has been that although we've, hypothesized or always thought that each of those cell types that we can see so clearly by microscopy would be distinguishable in terms of its transcriptome from the get-go, that's just not true. They're mainly shared gene expression profiles until you get into this the beginning of meiosis stage. So another thing that I thought would be of interest to Patricia and other people that think about chloroplasts is, even though our single-cell RNA-seq library construction method has a poly-A selection step, we get a lot of plastid transcripts and mitochondrial transcripts coming through the protocol. And I already told you that the endothecium is the only cell type that makes chloroplasts, and nearly all of the plastid transcripts map onto the endothecium, and with a tiny amount in this early zone, which we assume might be proto-endothelial cells. And then we have other transcripts that we've been very interested in, like the glossies that are thought to be epidermal markers, but the tapetum also, or a subset of the tapetum also secretes wax to make part of the pollen coat. So as we test these different marker genes, we're finding out, I would say, new things about some cell types that are very, very interesting. Okay, so a kind of final analysis that we do when we, when we think we understand a particular cell type is we make a heat map by stage. So these are the various stages of anther development and their approximate length. And we can look for changes in gene expression that are dramatic at particular stages. And actually, we rely a lot on the pseudotime velocity. So this tells us that as the tapetal cells are born, which occurs right here, they begin to express things, and then there's a strong blip as they're definitely separating from other cell types, but this is very few actually marker genes. But during meiosis, they're making lots of new gene expression to make components of the pollen cell wall. Then after meiosis, their transcriptome doesn't much change till near the end of their life. The tapetal cells eventually die by programmed cell death and dump all of their final contents onto the developing pollen. And here there's a huge amount of activity. This is in contrast to the endothelial cells, which we see from the very beginning express a very large number of transcripts that are associated with plastid development. And then after that, they just maintain those plastids until very late. So this is about 10 days, 8 to 10 days before pollen shed, when they finally become fully photosynthetic. There's a new burst of gene expression. So we're using these kind of analyses to decide what stages are actually meaningful, you might say, for particular cell types, in terms of where you might focus your attention on the expression of new transcription factors. We've become very interested in RNA turnover because transcripts do disappear. And so sort of opening new doors for future investigation. And the final thing I'd like to point out is I've made a big deal about using marker genes. And actually, one of the fruits of doing the single cell work is you get hints about new marker genes because you have things that are super highly expressed in a particular cell type. Myocytes, we have some new genes for the tapetum, although they're not as highly expressed as we would like or as highly differentially expressed. And then for other cell types, including our other somatic cluster 1 and 2, we're starting to pull out things that we can either mutagenize or hope someone else is mutagenized, and we would see a phenotype in that cell type. So this is also an important future direction and a really satisfying aspect of doing single cell RNA-seq work. You use what you already know to identify cell clusters. You use the cell clusters to get new marker genes to move forward. So conclusions from what I've tried to communicate today: single-cell RNA-seq is a valuable approach for understanding cell fate specification and differentiation, including the timing of events. Our new protocol is for us, super major improvement for analyzing plant cell samples. We think we're getting really close to the actual mRNA composition of the cells in situ, and we're recovering all the cell types in the expected proportions. So you can really study the whole organ. Two of our main findings are the major transcriptome reorganization in myocytes at the leptotene or leptotene-zygotene transition, and that haploid gametophyte development uses sporophytic transcripts until the binucleate stage. Then for our anthersomatic cell types, we're seeing how they acquire new properties over time. Based on Microsky and mutant analysis, we had hints of this, but now we have the underlying transcript information to better understand, especially sequential redifferentiation, which is a hallmark of the tapetum, which I would say is our major cell type under study presently. Now I'd like to add one more thing, sort of an advertisement for my university. Oh, well, first, who did the work? Here's Blaine in his natural habitat. His PhD thesis was the genome of a fern. So here he is in a little fern forest. And Brad Nelms, who was a former postdoc and now at the University of Georgia. So these two people really did the bulk of the work that I presented today. So the ad for Stanford, if there's anybody who's a potential PhD student in the audience, or if you're an advisor to a master's student or an undergraduate, my department is giving a preview. It's a webinar program with workshops on how to apply. So this is for people thinking of applying in fall of 2022, so soon. Our deadline is in late November, and you're considering applying to our PhD program. You need to be a current senior or rising senior undergraduate, a relatively recent college graduate, or a master's student. And importantly for my audience today, we really encourage international students. You're eligible for this program. It's basically how to apply to our program, also learning a lot from current graduate students about life at Stanford, about housing. You get a fully paid financial aid package. Is it sufficient to live in Palo Alto? There's some argument about that, but you'll hear the ins and outs of how students make do here. You get one-on-one feedback on your resume and personal statements. You can network with current students, postdocs, and faculty. So we really think that it's an important activity for getting involved. And it's just, in 2 months from now, it's the week of October 17th. This is kind of a long URL, but you can also go to the biology department at Stanford webpage, and there's information there, or send me an email and I'll send you this link. So with that, just say thank you and find out if there's any questions.
Thank you very much, Jeannie. And I think we have time for one question or so.
Okay. I'm such a slow speaker. Sorry.
Questions? I think Arnaud is in Zoom. Arnaud?
Yes, thank you very much for this talk, full of conceptual and technical breakthrough, I think. I have a question that is, does the step of sporophyte to gametophyte transition is related to a developmental change, in the tassel extruding, in terms of hypoxia, in terms of light? Do you think that there is this kind of big change related to an external signal?
It could be, because in our genotype, which is inbred Wisconsin 23, the tassel is visible. So you can see the tassel at the top of the plant more than a week before pollen shed, I would say like 8 to 10 days. And we know that's approximately coincides with the timing of the mitosis, the first mitosis. The trick about the tassel that I didn't go into is there's actually 7 days worth of developing anthers on one tassel. So you could say everything I told you is about the most advanced anthers on the tassel. So after the tassel is fully visible and like is unfolding and the oldest anthers are getting ready to shed pollen, say 2 or 3 days before pollen shed, the youngest anthers on that same tassel are just now undergoing the first mitosis. So I don't think it's not an all or none event throughout the entire tassel. So I don't think that something like light per se or emergence from the plant being fully exposed to the air could be a sufficient trigger, because the anthers are of so many different developmental stages, basically a week of development. So I guess that's like thinking that way is probably why we prefer to ascribe it to something that has to do either with the preparation for the mitosis, or perhaps mitosis itself, like the synthesis of a new round of DNA, erases or changes the epigenome in a way that can activate transcription. There is transcription during the mitotic cycle. So it's possible that it's even during mitosis or immediately after mitosis, as the two nuclei separate. If it's true that the vegetative nucleus is responsible for most of the transcripts, maybe that's what activates the vegetative nucleus to begin transcription. So we think our timing is, kind of plus or minus 8 hours, the way that we do everything. And we need closer timing. And probably the trick that we'd use is to label with EDU, which is a base analog that's taken up during DNA synthesis, so that we could score whether a particular cell was at the beginning, before, beginning, middle, end, or post-mitotic, and then do the transcriptomes.
And also, would it be difficult to check that on the female side, if this transition also occurs?
Yeah, I guess I've always thought of dissecting things out of the ear as much, much more challenging. I think everything I described would be feasible, just a lot more arduous to get the initial sample. And of course, there's more mitoses on the female side. So more possibilities.
Thank you.
Yeah.
Well, thank you, Jeannie, very much. I have some questions, but I'll ask you later. And
okay.
If somebody else wants to ask her a question, you can direct that to her directly, and she will answer probably. Thank you very much for your time.
And congratulations, everybody. 40 years is a long time.